Thank you very much. Thank you. Can everybody hear me? So you need me to use the microphone. So I appreciate the invitation. I appreciate your willingness to really be engaged in this type of activity. I first heard about this program just a few weeks ago, maybe a month ago, and I was proud to travel down here and speak to you today to engage the next generation of creative genetic minds with a focus on cancer and oncology. So just a little bit about myself. I have a focus on genomics and functional genomics with respect to oncology, but more so with a drug discovery perspective, with the goal of translating cancer genomic information to actionable clinical endpoints. What can we learn from cancer genetics? What can we learn from sequencing the cancer genome? And how can we leverage that data to develop the next generation of targeted therapies and reduce mortality across a number of different cancers. I trained at Dana-Farber Cancer Institute and Harvard Medical School in Boston, Massachusetts with Linda Chin. There, again, my focus was on genomics and functional genomics. I then moved on to what is still called the Belfer Institute for Applied Cancer Science, where I led target discovery approaches, utilizing functional genomics to identify novel targets in a number of different cancer types. As you were just told, a number of the platforms that we were able to build successfully led to a number of drug discovery programs with Merck Research Products, Research Laboratories, and Sanofi Aventis, two of the largest pharmaceutical companies. In September of 2011, we were recruited to MD Anderson Cancer Center in Texas, where we started a similar institute called the Institute for Applied Cancer Science. Again, this is more heavily focused on internalizing the drug discovery efforts and really accelerating the discovery of novel targets and the development of effective drugs. So what I wanted to do today, I know that the past couple of days you've been sitting through some exciting lectures. And this will continue tomorrow and I believe on Friday as well. And so unfortunately, my colleague Andy Futrell could not be here today. Andy is really a pioneer in the field of genomics, but due to travel issues and weather issues in Houston yesterday, he was unable to fly in. But what I'll try to do is try to touch upon a lot of his efforts, along with Linda Chin's efforts and many others in the field of genomics, as a lead into some of the work that I'm going to present today. So I thought I would split this up into two sections. The first being more discussing the challenges that we face in oncology and developing novel therapeutic drugs or effective therapeutics. And this really has to do with the complexity of cancer. Unlike cardiovascular disease or diabetes, cancer itself is a term that essentially categorizes hundreds of different diseases. Breast cancer is not a singular disease. There are many different subtypes. There are many different genetic drivers of the disease. So in essence, we're fighting an incredible battle. There have been some successes in the field of oncology with respect to personalized medicine. I'll talk about a few of these. But there are many failures. And again, it is a challenge, and I'll talk about that through the first part of the presentation. And then really a focus on personalized medicine. We are leaving an era of cytotoxics. These are cancer drugs that are effective, but yet will target every cell in the body, and therefore you see the systemic side effects associated with cytotoxics: alopecia, nausea, vomiting, and associated with these is a really decrease in the quality of life. And so as we enter the genomic era and personalized medicine, I think what you'll see from this presentation is that we do have a number of different activities that we can expand upon. I'll talk to you about, introduce you to the field of functional genomics and how we use functional genomics to translate these data, again, into actionable clinical endpoints. The idea that sequencing is just simply not enough and that there has to be some biological weight of evidence. And then I thought at that point we'd take a break. And then when we come back and reconvene, I'll introduce you to the Institute for Applied Cancer Science and give you a brief 40-slide introduction to the process of drug development, from target identification all the way through late-stage development and preclinical proof of concept, and how we go about this at the Institute for Applied Cancer Science to accelerate this process in an academic setting. And then lastly, you know, I'm happy to have a question and answer session, and I know that I'll be joining you for lunch today as well. So we'll go right ahead and start. When you think about cancer and drug development in cancer, and again, I apologize. I provided these slides last week. There are a number of slides that are not included in your deck. I will certainly send the updated copies, but a number of the slides that you have are included. Just maybe switch the order around a little bit. But this is really speaking to the hallmarks of cancer. About 12 years ago or so, Bob Weinberg and Doug Hanahan took the Herculean effort to review cancer literature and inform on the hallmarks of cancer. These phenotypes that are conferred upon cancer cells that provide them the ability to grow with independent of mitogenic stimulation, to evade the power of our own immune system, to evade apoptosis, to induce angiogenesis, to provide them oxygen in a blood supply, to metastasize to distant organs, and then essentially replicate and provide that infinite lifespan. So these are all hallmarks of cancers. Not every cancer or a solid tumor or a blood tumor has these individual phenotypes, but these are the phenotypes that we target when we think about developing effective therapeutics. And as you can see here, there are a number of different drugs that are currently in the clinic. EGFR inhibitors, which I'll talk about a little bit, CDK inhibitors that again inhibit growth. Really a pioneering effort of Jim Allison, now at MD Anderson, was the development of anti-CTLA-4 antibodies, which again allows us to reset the tumor to really harness the power of our immune system. Telomerase inhibitors, inhibitors of VEGF to inhibit angiogenesis, but also in the field of apoptosis and evading apoptosis is to think about pro-apoptotic mimetics of the BH3 proteins. So you can see here there are a number of different phenotypes that we necessarily have to focus on. But what I wanted to show you in this in the next series of slides is that, again, this is a disease that is incredibly complex. This is what's called an array CGH plot. This is just a representative plot. You know, what we have here on the y-axis is frequency. On the x-axis, these are each individual chromosome number from 1 all the way to the sex chromosomes X and Y. And what you see here is a shift in any direction, higher or lower, are changes in the structure of individual chromosome numbers or chromosome number variations between a tumor and a normal reference. And so you can see here there are a number of genomic changes. But in addition to the genome, in which, again, you were supposed to hear about this morning before my talk from Andy Futrell, there are a number of different efforts or activities that provide these phenotypes. Solid tumors and blood tumors, for that matter, are housed in this intricate microenvironment, which includes the host stroma. So there is this interaction between the host, the normal body, your normal body, and the foreign substance, which is the solid tumor, providing, again, protection, providing growth advantage, providing the necessary blood vessels to feed the tumor, and provide it that necessary mitogenic stimulation to grow. In addition, there is lineage context issues. A tumor that develops in the brain is very different from one that develops in the breast to one that develops in the colon. Again, we'll speak to this quite a bit in the later slides. There's also genetic context. There is hereditary cancers, but there's also germline associations. I think you'll speak to that tomorrow with Karen Liu. Heritable cancer syndromes. And then lastly, there's the macro environment. There's ultraviolet radiation, which is clearly a causative effect of melanoma. There's pollution. There's toxins. There are a number of things in our outside world that can promote or drive cancer development. And so this idea that cancer is complex is really an important issue to appreciate as we go through this talk. What I'm calling here is not necessarily genomic analysis, but rather omic analysis, right? So, and I'll show you this in a little bit when we get to what the TCGA has done. But what Andy was going to present is the world of genomics and where we've come. So more than a decade ago, when it was agreed upon that we should sequence the human genome, it took 10 years. It cost about $1 billion US. And we got a draft of the human genome. Today you can sequence the genome of multiple samples in parallel in just a couple of days for $1,000. So you can see how this technology has evolved. There are even companies out there, it's just like every technology. There's even companies out there that'll tell you we can sequence the genome in a matter of hours for a few $100s. That's where we'll be going. It's almost like the iPhone, or, before, I don't want to date myself, but when I was, I didn't have a cell phone until I was in college. And there was no text messaging, or now you can, this side of the room can text message that side of the room. That could be FaceTime or Skype, and this is the same technological revolution that we're entering in with genomics. That it's robust, it's high throughput, and it's informative. But when thinking about cancer and how we discuss personalized medicine, I'm going to give you one example, and that is in human melanoma. So when we think about cancer, there is a disease. And here I'm showing you on the top, this is a picture of a mole, a melanoma that has developed on the surface of the skin. On the bottom, there's the histopathology that are used by pathologists to show, yes, this is a cancer, this is an invasive cancer, and they inform on the grade and stage of that disease. To really inform, is this a primary lesion that could be resected, or is there lymph node involvement? Has the tumor spread to other organs, including the liver and the brain? And now once you have the disease, you need to inform on the pathogenesis of that disease. What is causing that disease? And so here on the top is a classic example, a seminal discovery by Andy Futrell and his group a number of years ago, in which they sequenced a number of melanoma tumors compared to associated normals, and what they found in 60% of those tumors was a recurrent mutation in the oncogene, the kinase BRAF. And so what they found is that there's this recurring V600E mutation in the T-loop of this kinase, which makes it constitutively active. Now this is a kinase that promotes growth. As shown on the bottom is just a very simplified schematic of this signaling pathway, where if you have activating mutations in BRAF, again, you're independent of mitogenic stimuli, you activate MEK and ERK, and you drive proliferation through the nucleus. Now this, again, was a seminal discovery. This pinpoints a driver of the disease. And now over a number of years, through the work of Gideon Bollag at Plexicon and now Roche, we've been able to develop an inhibitor against this particular driver allele. So this is vemurafenib. This is a compound that targets a very potent and selective inhibitor of BRAF. And what's shown on the bottom is called a waterfall plot. So this is through a number of years of preclinical studies. These are the results of the phase 2 trial. And what this is showing in this waterfall plot, 0 is considered baseline at the time of treatment. And then they measure changes in tumor size or growth over time upon treatment. And what you see here is a waterfall plot. If it's less than 0, it means the tumors are shrinking. Right? And so you can see here in just a small number of patients that there were incredible responses. in a BRAF V600E mutant patient population to this drug. So again, from disease, this is a human melanoma, to the pathogenesis of this disease. This is caused by a recurrent mutation in a kinase, to the development of an inhibitor against that kinase, to clinical activity. And so this is one of the successes of personalized medicine, where we went from genomics all the way through the clinic. So vemurafenib is FDA approved. It is standard of care for BRAF mutant melanoma patients. So this is one example, but there are a number of examples as shown here on how cancer genetics is accelerating the development of novel therapies and personalized targeted therapies, from target discovery all the way to clinical proof of concept. The first example is Gleevec, or imatinib. This is an inhibitor against a fusion called BCR-ABL. This took about 41 years, as it says here, to go from, again, disease pathogenesis, identifying the lesion that is causing the disease, to drug development. Now, over time, we've shortened that. As you can see on the bottom, BRAF inhibitors such as vemurafenib was from 9 years from the time of identifying the causative lesion in BRAF mutant melanoma to FDA approval in 2011. And then also the ALK inhibitor. This took about 4 years between identification of the critical EML4 ALK fusion in lung cancer patients to the FDA approval of crizotinib. And so this gives you an idea. And this is a few successes, but there are many failures, as I will tell you. But this gives you an idea of how we could harness cancer genomics to inform on disease pathogenesis, and then really enter these high-value, high-priority targets in a drug discovery machine. So this is really, this picture, these two pictures speak really 1,000 words. So on the left is vemurafenib. This is a paper study published in New England Journal by Keith Flaherty. He ran the clinical trial. This is one representative patient. This is FDG PET, in which they use a labeled glucose. And what you see here, there are a number of tumors that are lighting up in this patient, this full body scan. This is baseline. And then after treatment with vemurafenib, you can see an incredible response. This is one representative patient, but again, here's the waterfall plot, again showing nearly 60% of patients showed a partial or complete response to this treatment. Again, going from genomics to the clinic. On the right-hand side, this is a CT scan. This is a patient treated with crizotinib. So this is a patient at baseline. You can see the lung cancer is just shown here. Pre and post-treatment with crizotinib. Again, you can see almost a complete response. And so these patients were selected. So this was really hypothesis-driven or biomarker-driven clinical trials, where you select and stratify patients based on a particular lesion, the BRAF V600E mutation or the EML4 ALK translocation. So this is really what we're aiming to do, is to show complete response using an agent that is developed against a particular target that's driving a disease. Now, with that said, there are a number of drugs that fail. Now, greater than 95%, and this is stunning, first time I had heard this number, greater than 95% of the drugs that enter the clinic fail between phase 1 and registration. And that is just an astonishing number, considering that it takes 7 to 12 years to develop drugs, costs about $1 billion to develop drugs. And so we have to improve the success rate. That is clear. There are a number of other successes beyond vemurafenib and crizotinib, but there are many failures. So this was a Nature review and drug discovery paper, which showed that, so they looked at 107 drugs, not just oncology. About 30% of the drugs that they studied were oncology. But they looked at 107 drugs that entered phase 2 clinical trials, right? So phase 1 trial is dose escalation, maximum tolerated dose, safety, making sure that the patient, there are no overt toxicities. Phase 2 is looking for efficacy in a defined patient population. Phase 3 is the pivotal trial, where you're comparing a new drug to that of standard of care to see if there is any survival benefit. Now, of the 107 drugs, 89 failed in phase 2. And now this pie chart is essentially showing the reasons for that failure. So one, there's strategic issues. There are safety issues, so there was toxicity observed. Pharmacokinetics or bioavailability, we'll talk about this a little bit later in the talk. But PK or pharmacokinetics, that's understanding what the body does to the drug, right? And then lastly, 51% of all the drugs that failed because of efficacy, suggesting that we're treating the wrong patient population, we're treating the wrong disease, or we don't know enough about the disease. So these patients were treated with a drug, and there was absolutely no benefit. And so we need to better understand, and the word that you'll hear me, speak of, pretty much throughout the talk, is context. Context in the sense of genetic context. What are the background cooperating mutations? Lineage or cellular context. Treating, and I'll give you an example, but BRAF inhibitors in melanoma are very effective. But if you use BRAF inhibitors to target BRAF mutant colorectal cancer, it's completely different. They're ineffective. And so again, a cancer on your skin is very different from a cancer in your colon, even though they're driven by the same mutated allele. And so we'll speak a lot about context. But that was really what drove us to really focus our efforts on understanding context. If you if you identify your targets in the context of a particular genetic allele, you have your clinical path hypothesis, or you have your phase 2 trial already built in. And so this is really, the focus of our institute. It's really the focus of the field right now. That it's not necessarily about the drug. It's not necessarily about the target, where you have a target in which you have function. You identify the target through patient omics. This could be genomics, but many others. You've validated that target in a number of preclinical models. I'll give you a few examples on how we do that, what our stringent criteria is. You have the mechanism of action. You know exactly what this target is doing. You know exactly what happens when this particular amino acid is mutated. And so that gives you the right target. Right? That's the first step. And then you build a drug against that target. Here, you have small molecules or large molecules, biotherapeutics, antibodies. You have a number of assays. And this is really the second part of the talk, is to go through the process of building a drug. And how do we do that? It's not simply doing a one-off chemistry experiment and saying, all right, I now have a drug, I'm ready to go into the patient. Clearly not. There are a number of assays that we use informing on the biology and mechanism of action. So here we're going to use chemistry to inform on the validation of this target. Is it required for the growth of that particular tumor? But at the same time, once you have the right drug against the right target, it's informing on the patient population, the right patient. The right target, right drug for the right patient. And that's through biomarker-driven trials, right? One example that I will give you later today on context is that if you take a BRAF inhibitor and you treat a BRAF mutant patient population, 60% of those patients will respond. If you treat the other 30% of melanomas that harbor an NRAS mutation, it is actually worse than standard of care. Nobody could have predicted this, that it actually potentiates the disease. I'll talk a little bit about this later. So informing on the biomarkers, predictive biomarkers, how do we anticipate response? And how do we stratify a patient population? How would we, in this group, how would we identify the 10 people that would likely respond and the 70 or so that wouldn't? This is instead of taking all comers, and then you're really minimizing your effect. And so that's really the effort that we have here. Talking about drug-drug combinations and rational drug-drug combinations. What happens when you treat a cell line with a particular inhibitor? What are the mechanisms of resistance? And you'd be amazed at how cancer cells evolve. So this is Darwinian evolution at its finest, right? Cancer cells confer their own ability to evade a specific inhibitory signal. And we know that. Crizotinib and vemurafenib, incredible successes. but it's not durable. So within 8 months, 12 months, these patients relapse from those particular tumors. We're starting to inform on mechanisms of resistance, some of them that I'll talk about later in the talk. So understanding what those mechanisms are will help inform on drug-drug combinations. What 2 drugs could we treat our patients with to inform on efficacy? But this is really a new era of clinical trials where it's not everybody goes into, one group. We split the room up. You're getting drug, you're getting placebo. It's evidence-based, predictive biomarkers. All right, of all of 70 of you, 10 of you have the biomarker, you're going to get drug A. 20 of you have a different biomarker, you're going to get drug B, and so on. This is really the new era of trials. And this is one trial that's being performed at MD Anderson called the BATTLE trial. This is biomarker-driven in lung cancer, and it's been incredibly successful. And so with respect to functional genomics, how can we use functional genomics to inform on context? And this is really what's so critical. Where you have a patient population, as shown here up on the top, you have 4 patients. These 4 patients, through the efforts of the Cancer Genome Atlas and the International Cancer Genomics Consortium, again, this is a lot of work that I anticipated Andy talking with you a little bit about today, so when we get to that section, I'll take a little bit more time. But those four patients and the tumors that have been resected from them will be annotated on multiple omic levels. We're going to sequence their entire cancer genome. We're going to inform on a number of different cancer-related phenotypes. This effort here just provides us lists. And you'd be amazed. I showed you one CGH plot, but I think in the next couple of slides, you'll see just how complex the problem is, where we get lists that are hundreds to thousands of genes long. And so how do we look at a list in an Excel file and say, all right, this is the one. This is the one we're going to invest $1 billion on. This is the one that if we create a potent drug, we are going to cure this disease. It's impossible. No big pharma, with all the billions of dollars that they have, would be able to take on a drug discovery portfolio that's 1,000 genes. And so what we do is we pass this through a filter. We functionalize or translate that genomic knowledge using a number of different functional genomic platforms. And what the platforms that we have built at MD Anderson have allowed us to inform on context-specific targets. Again, in what genetic or cellular context is that target rate limiting? Because that'll inform on, again, the right target, the first step in drug discovery. We'll inform on mechanism of action, clinical path hypothesis. What's your path to the clinic? What's your phase 2 trial going to look like? Again, biomarker-driven. And so the goal of this is to stratify patients, just as I had used it as an example, where of these 4 patients, 3 of them are going to get treatment A. They have one biomarker. 1 of them is going to get treatment B. In the old days, all 4 patients would get the same treatment and maybe only suggest, as an example, hypothetically, if all 4 patients got treatment B, your success rate would only be 25%. And that's what we're seeing. We're seeing very little efficacy. And so we need to be more biomarker-driven, predictive. And so when we think about the barriers to this progress, and what are the challenges we face? There's an elemental knowledge of cancer omics. Again, it's not necessarily genomics. I know this is a genetics course and a genomics focus. We can sequence the cancer genome, but it's everything else that we're doing. We're integrating these activities. So we're sequencing the genome. We're informing on proteomics, where we're studying proteins. We're studying signaling molecules. So what is the functional consequence of this particular mutation? Is there activation of one pathway or another? We're talking about metabolomics, metabolism. You often hear that in the news about the next diet drug, or this will increase your metabolism or whatnot. But cancer cells have their own metabolism, and it's very different from normal cells. Cancer cells, through work over 100 years ago by Otto Warburg, a Nobel laureate, had shown that cancer cells consume glucose more than a normal cell, sugar. And so they have a completely different metabolic program. And so again, understanding the field of metabolomics. We're using mass spec-based technologies to inform on surface proteins that are expressed on cancer cells. We're informing on circulating tumor cells, those cells that shed off the exterior of the tumor and are floating in our bloodstream. So there is really an integrated effort to really inform on every aspect of the cancer cell to give us the best chance to succeed. Limited insights into the factors driving cancer. Again, it has to be genetic and functional weight of evidence. We're getting lists. We're getting, okay, through this integrative analysis, here are the genes that we think are high priority. Now, even through the analysis using human and mouse, multi-level data, we're still getting lists that are hundreds of genes long. And so again, we need to inform on which genes are actually functional and critical. A poor understanding of the target's biology. Again, context, cellular and genetic, and we'll talk about this quite a bit. And then lack of insight on appropriate drug-drug combinations. We know that tumors will find a way to bypass a point of intervention. We've shown that in the best drugs available. Imatinib, Gleevec. Mortality through BCR-ABL translocation has dropped significantly since the development of Gleevec. However, one mechanism of resistance is a single point mutation that now makes that oncogenic kinase resistant to that particular drug. Now that we've informed on that mechanism of resistance, we now have second and third generation BCR-ABL inhibitors that are entering the clinic that are showing success. And so when we think about genomics, and I'll touch upon these 4 challenges as we go through, but when you think about genomics, it really has evolved. Just like the technology that we use with telephones and whatnot. But through what we call massively parallel sequencing. So in parallel, massive, meaning large number of individual sequencing reactions, we can comprehensively characterize the cancer genome. And so we can put in genomic DNA from 500 to 1,000 different tumors. We can put them on our sequencer. A couple of days, again, we have a full copy of the human genome with respect to that particular cancer. Now, what we're informing on are the individual types of mutations. There are point mutations that I'm sure you've heard about in your genetics courses. So these are single nucleotide mutations. Some may be what we call silent mutations, where you don't change the amino acid code. Some may be nonsense mutations. Some may be frame shift mutations, where you may have a deletion, or you add an extra base. Causes a frame shift in the entire protein, usually a premature stop, completely kills that function. We're informing on deletions, homozygous deletions. We see this a lot in what we call tumor suppressor genes, such as P53, which you heard from Gigi Lozano yesterday. PTEN, another classic tumor suppressor, where there's mutations, but there's also deletions, where we'll delete an entire, in cancer, you'll see deletions in an entire segment of a chromosome. And I'll show you that in just a few minutes. Chromosomal gains, things like EGFR. This is a receptor tyrosine kinase that's commonly amplified in cancer. Well, you see that segment of DNA, again, I'll show you in a few minutes, is amplified, where instead of having 2 copies in your genome, you may have 10 copies. And through that dosage effect, you now have an oncogene that is promoting growth. But with respect to EML4-ALK, you have translocations. And so being able to sequence both DNA and RNA that come from tumors, we can inform on all of these different events in a comprehensive manner. So the translocation breakpoints. So you're talking about one gene on chromosome 1 is now fused to a gene on chromosome 10. Now, the gene in chromosome 1 might be regulated very different and may be constitutively expressed. And so if that's fused to an oncogenic kinase, again, you can see constitutive activation of that signaling molecule. The EML4-ALK fusion is actually due to an inversion on chromosome 2, where you have a segment of the chromosome that has just flipped. And because it has flipped, now you have produced a fusion protein that drives 5% of non-small cell lung cancers. So again, using these type of approaches and these technologies will help us inform on disease pathogenesis, and then provide us, again, that first step to the next therapeutic. But cancer, I'm sure, and my textbooks are the same. Throughout this slide deck, you'll see the same. You'll see signaling pathways where kinase A phosphorylates kinase B, phosphorylates kinase C. But the reality is, the textbooks are simplifying this process. Signaling pathways are not linear. Cancer is not just pathway A, B, and C, and then if you stop them in one position, you're going to cure cancer. Cancer is associated with a myriad of mutations that cooperate. And so I see this as, this spaghetti, a lint ball, whatever is your fancy. But the reality is, cancer is incredibly complex. This is what we face, where in a single cancer, you may have hundreds of mutations. Of those 100 mutations, there might be 5 critical mutations, and they're all cooperating to drive tumor growth. We know that. So in BRAF mutant melanoma, 80% of benign nevi that are on our skin harbor BRAF mutations. That's scary to think, right? But of those 80% of nevi, they're benign. They're not tumors. And so there has to be, through the evolution of that disease, you acquire additional mutations that cooperate with BRAF, right? To turn this into now a malignant disease. But this is the reality. This is what we're seeing here. And so in this slide, what I wanted to show you, this is an older slide. This is, and again, I apologize. Every time I flip the slides, I see everyone. I apologize for. Just want to make sure you're staying awake. If you keep flipping, I know you're listening, I guess. But I apologize for that. But so this is a slide that, again, this is array CGH, comparative genomic hybridization. This was one of the earliest technologies that allowed us to look at chromosome number variations using probes that spanned all of the chromosomes. We could show, compared to a reference, if a genomic segment was enriched or reduced in a tumor compared to normal. And so again, on the y-axis is individual patients. On the top is individual chromosomes. If you see blue, it means that genomic segment is lost compared to normal. If you see red, it's gained. And so first, ovarian cancer, you can see, is a complete mess. A genomic nightmare. Right? And ovarian cancer is one of the most deadly cancers out there. We have standard of care, platinum, platinum-based drugs, Taxol, very effective. But many patients relapse 5 years after treatment. Many patients. Ovarian cancer, again, through this evolution, is a disease, and there's reasons for this. Ovarian cancer is asymptomatic. You don't necessarily know you have ovarian cancer until you're stage 4. You usually come in with abdominal pain, right? So there's no primary disease that we can identify. And so that's, again, a separate talk about, imaging and how can we predict. But if you look at this path, compare ovarian cancer to glioblastoma. Glioblastoma is a brain cancer, right? There are very focused regions in GBM. If you think of ovarian, you see these recurrent red lines here on chromosomes 3 and chromosome 8. So on chromosome 8, you have this recurrent amplification or gain in 8q, right, of this particular chromosome. Now this gain harbors the oncogenic transcription factor MYC. It is one of the undruggables, like KRAS. It's an oncogene that nobody has been able to drug. But no, if we could drug MYC or RAS, we would have incredible efficacy in the clinic. And so again, it's something to think about. Glioblastoma its CGH profile is relatively benign. You see loss at chromosome 10, whole chromosome loss. Within that region is the tumor suppressor P10, right? And so you can see cancers, they've evolved with this ability to know exactly what they want to amplify and what they want to delete. Breast cancer is very different to colon cancer and lung squamous, right? On GBM, on chromosome 7, the amplification is the gene product EGFR. It's an oncogenic RTK. Drives proliferation independent of mitogenic stimulation. So you see, every cancer is different, and this again speaks to context. Ovarian cancer is very different from lung squamous. KRAS mutant lung squamous is different from EGFR amplified lung squamous. So you can see when we talk about cancer, it's not one disease. Many diseases within diseases, and so on. So it's almost exponential. So this was really one of the present, and I'm sorry this is not in there. Man, this is going to happen all day, right? But I will give you an updated form of this. But this was a study that Andy Futrell would have presented this morning. Again, one of his many seminal discoveries. So he worked with a group. This is a renal cell carcinoma. So this speaks to tumor evolution, and again, what we're up against as biologists, geneticists, and whatnot. And so what they actually did is they took a renal cell carcinoma and they took a biopsy. So this is a tumor. And as you can see, they took individual biopsies from around the different tumor. And if you look at the schematic, as shown up here, they also took biopsies from a number of different metastatic sites. M1, M2, M2A, 2B. As you can see here, there's a chest wall mets. There's a metastasis just right outside of the kidney. And then what they did is they did deep sequencing. Again, using massively parallel sequencing, the evolved technologies. And then what they started to do was to start informing on tumor evolution. Not necessarily what's different between a primary and metastasis, but they built a phylogenetic tree. Again, speaking towards Darwinian evolution, where it starts with a common tumor suppressor mutation that's called VHL. Now, as you progress, you start to see branches that evolve. You start to see now in 4B, which was taken from this area up here, has a different genetic profile. So it branches. The metastases have a different genetic profile, right? And so you see this branched evolution effect. Again, so even within one mass, one solid tumor, we see very different genetics. We see very different cooperating alleles. As he's shown here, you have a missense mutation in mTOR. You have activating mutations in SETD2, which regulates the expression through epigenetic modification of genes. And so you can see through this that cancer continues to evolve. And to me, this was one of the scariest papers that I had read, because not only is cancer complex at the genomic level, but within a patient, within one solid mass, it's very different. So cancer is a heterogeneous disease, both at the genomic level, but also on the pathological level. So these are the things we have to keep in mind. It's not necessarily going to be one driver. So this is really speaks to the reality of the disease, right? Drug development in a textbook would suggest that you have an inhibitor which inhibits an enzyme which catalyzes the reaction of a substrate, S, to a product, right? And that if you inhibit that enzyme, you inhibit that product, right? But it's just not that simple. In textbooks, again, and even through my talk, you'll see very linear diagrams. But it's not that simple. The reality of the disease is that there are many ways to activate one node. So if you inhibit this, should you inhibit that? Well, not so, because this kinase can actually activate that. And so this is what we need to face. So this speaks to understanding cancer beyond necessarily just the genome. Understanding the signaling molecules, understanding the metabolomic profile, metabolic profile. A number of different activities. So it's a complex disease. And so that really brings us to, how are we as a community addressing these issues? And it starts with an integrative effort, TCGA and ICGC, building an atlas. So we have an elemental knowledge of cancer omics. And so what we're starting now, what has started a number of years ago but is now fully integrated, is a comprehensive approach to understand cancer. And so through a number of years ago, TCGA was started, the pilot projects of GBM, ovarian, and lung. The idea that they would sequence, 500 tumors. And that has been informative. Through those data, we've been able to inform on disease pathogenesis, cooperating mutations, pathways that are activated more than others. But since then, we've gotten into the area of epigenetics, focusing on DNA methylation. Methylating the promoters of genes could shut them down, turn them off. If you methylate a tumor suppressor gene, again, you remove a break or a checkpoint against tumor development. Genotyping, again, at the sequence level, through mutations, amplifications, and deletions. Expression, looking at the expression of particular genes. You know, is there copy number-driven expression? I showed you CGH profiles where regions of chromosomes are amplified. Now, does that translate to overexpressed proteins, oncogenes, or others? Associating that with functional proteomics. Again, this idea of combining or integrating sequencing, looking at deleterious mutations, and then informing on the function of those mutations. You know, if you have a mutation in kinase A, does substrate B activity go up or down? And so that's the idea, that we need to inform from genomics all the way through proteomics and others. We're looking at tumor phenotypes, looking at the effect of stromal cells. You know, not only is the genetics of cancer very complex. There's one disease, pancreas cancer, a very deadly form of cancer. There was a study that informed from a mouse model that there's actually a stromal barrier in a mouse model of pancreas cancer. That the host stroma, the cells that we rely on and call ours, form a cap. And therefore, drugs can't penetrate that tumor. You know, so the microenvironment is critical. So understanding the relationship between tumor and stromal cells, the influence of the immune system. And then we need to integrate all these data in this Circos plot below, which can inform on amplification and deletion, mutations, rearrangements. You'll see a number of these plots throughout. But this is what the TCGA and ICGC are doing now. They're doing this for 50 different cancer types. They're increasing the number of tumors. But again, we're at really the tip of the iceberg with respect to genomics and omic profiling. But then it comes to, you know, the factors that are driving cancer. Again, understanding which ones of all of these genes are important. And so this is really, when you think about translating the cancer genome, it's multiple segments. You have cancer omics. So you're getting omic profiling, as I just told you. Genomic, epigenomic, proteomic, etc. And so this is the first step. Then those have to be, you have just, I don't know, buckets of data that come off these machines. And then you have to analyze them. And these are using sophisticated platforms, sophisticated bioinformatic and computational biology to prioritize those that are likely more important than others. And this could be based on statistical significance, you know, p-value, recurrence. This mutation is observed in 10% of all patients that were sequenced. To me, that would be something that I would prioritize. We are now able to prioritize mutations within a gene. So you have one gene. You may have 30 different mutations. We're now able to predict using algorithms in silico on a computer which ones are likely more important than others. Is it perfect? No. But it helps us prioritize. So instead of working on 3,000, we may only have to work on 500. Makes it a lot easier. But then there's function. Once you have your gene lists, function. So performing functional genomics to assign disease relevance. Again, in a particular genetic or cellular context, which one of these genes that are mutated, amplified, deleted, rearranged are important. And then lastly, through these studies, you inform on insight, disease pathogenesis, deep biology to inform on mechanism of action. Which one is important? What is the effect of inhibiting that target? And can we do it pharmacologically? So these are how we prioritize targets. And that is not just necessarily genomics. And so when you think about what the TCGA and ICGC and through the evolution of these technologies, the type of information we're getting, right? And so this really speaks to the challenge of translating genomic data into drug development endpoints. Our focus as an institute is to develop drugs. Drugs that are safe, effective, novel, biomarker-driven trials that'll inform on efficacy. And so you need to complement structural characterization with functional annotation. As I was saying for the last 45 minutes or so. You have hundreds to thousands of candidates. You have drivers versus passengers. So you have a driver, as many commuted to campus today, that is controlling the vehicle, right? You are determining which way, how fast, when to stop, when to go. And then you have passengers, potentially. You may have had somebody, a companion in your front seat, back seat. And so when you think about cancer and drivers and passengers, you have those that are of critical importance, those that are driving the growth or providing that selective advantage of cancer cells. Then you have passengers, those that are biologically inert, neutral. The person in your back seat falls asleep, you're still driving the car. So they're not necessarily required, right? And so drivers versus passengers, we're focused on those that are absolutely mission critical. If the driver falls asleep, the passenger in the back seat can't necessarily take over how that car maneuvers and functions. And so within the cancer genome, there are drivers and passengers. There's relative importance of one driver from another. You may have multiple drivers. I can't come up with an analogy with respect to an automobile, so, but you may have a number of genes that contribute to this, to the growth and development of a tumor, right? And so now you need to inform on which one is the most mission critical, which is the most important. And what happens when you inhibit that? Do the other drivers, take over and help evolve that cancer? Again, context-specific actions and the deep biology to inform mechanism of action. So this is a study that we published in Nature earlier this year in which we sequenced the genome of 25 human melanomas and matched normals. This is not in there either. Sorry. Sorry. And so what we did here is we sequenced the genome of 25 melanomas, as you see here through these Circos plots. So this is just a viewer that allows you to look at comprehensive data from this analysis. So this is a wheel. So you have chromosome 1, 22, and then the sex chromosomes. You can look at individual point mutations. You can look at amplification deletions all around the wheel. And then you can look at translocations. So what these purple lines represent is there's a translocation between 8q and 1q in this particular patient. You have translocations between 3 and 15, right? And so then you can see, even from these 4 representative patients, that they're all called melanoma, but clearly their genomes are completely distinct. You have some, not quite sure what's going on here, but MEO32, you have all kinds of recombinations that are ongoing, right? And so we looked at all of this data. We came up with a recurrent mutation in a gene that's called PREX2. Function unknown. There was some publications suggesting that it regulates PTEN, that it might be a tumor suppressor. And so what we did is we found a number of mutations. In this one gene, we found all of these mutations that are listed. So this is the amino acid G106E, right? And so some of these are more deleterious than others. Some of them are probably less so. A proline, 665 to a leucine, right? It's not necessarily an amino acid change that I would be concerned about. it's the changes to glutamic acid and aspartic acid. Those are phosphomimetic amino acids. Those are amino acids that make a kinase believe that it has been activated, right? That it has been phosphorylated. So these are some of the mutations. But what we found, using one of these algorithms that allows you to predict which mutations are more functional than others, we put all these mutations into a computer program. And it said, okay, some of these mutations, as shown in red, are more important. They're more deleterious. They have more functional consequence, right? Because it would have been very difficult for us. It wouldn't have been difficult, but it would have taken some time to engineer all of these mutations, express this protein, mutant protein, and determine what its function was. And so we picked the 6 here. You have a number of G106E. You have a number of premature stops. So these are termination mutations, where you go from a lysine at position 278, and that lysine becomes a stop codon, right? And so therefore, you make a smaller gene product. And so you cut off 75% of the protein. And so what we did is we engineered these 6 mutations. We expressed them in a melanocyte, and we let the mouse tell us which ones were important. I'll talk about this a little bit more later, but what we did is we engineered these cell lines using lentivirus technology. Are you familiar with genetic engineering of cell lines? I'll take that as a no. But, and please, I probably should have said this, feel free to ask questions throughout this. This doesn't have to be a lecture. I feel like I'm in college again. I can see it. I can see myself in your same seats wondering when's lunch. But please, when's the break? When's the bell going to ring? But please, let's make this as informal as possible. But so there have been a number of protocols that have been built, technologies that have been built over a number of decades that allow you to genetically manipulate cell lines, right? Using virus, live virus. So lentivirus, right? You can package anything in a virus. So just like HIV, right? Human papillomavirus. You can take a lentivirus, and you can package within that lentivirus a DNA of your choice, right? A plasmid of your choice that expresses whatever you want to express. You put it into cells, you add all the components that make up a viral capsid, and now you have live infective virus that within it has your DNA of interest. You sprinkle this virus on top, I'm simplifying this of course, you sprinkle this virus on top of cells, the virus attaches, it gets ingested into the cell, it releases your components that are now integrated into the genome of that cell line. Right? So this is lentiviral technology, and this is going to make more sense later when I talk about how we're using these type of approaches to manipulate cells. And so this is one approach that we took. We engineered cells to express all of these mutations. We took these cells, we expanded them, and then we injected them into the flank of a nude mouse that has no immune system, right? No immune system that can fight off that foreign pathogen or that human cell line that's growing now on the back of its flank. And then we let the mouse tell us which ones were important. These were tumor-naive melanocytes. You inject them in the mouse. They typically don't form tumors. You're talking 10 weeks, only 2 mouse formed a palpable nodule. But if we take that same cell line and we express the K278 stop mutation, you see now 80% of these animals form tumors. So we've in fact taken a benign melanocyte that expresses BRAF, that doesn't form tumors, injected it to express, engineered it to express a gene with a cancer-relevant mutation, and now it becomes a cancer, right? This is not magic. This is not something we invented. This has been going on for years, years and years. But what it allows us to do is functionalize this genomic information. 25 melanomas. We have a recurrent mutation in PRX2. 14% of patients had mutations in this gene. And now, using engineering and functional genomic approaches, we've been able to inform on which mutations are absolutely critical. We've gone on to show that the function of these mutations and the mechanism of action is very interesting. That's the next paper that's coming up. So again, going from an exercise, we have 25 melanoma tumors. We're going to do whole genome sequencing, low-pass whole genome, high complexity, high coverage whole exome, RNA-seq. We're going to inform on translocations and mutations. And we're going to show function. Not all mutations work like this. As you can see, some of the other mutations, such as the P948S, really didn't do much beyond what the wild type did, or what our control did. And so you can see not all mutations are equal. They're not all deleterious in nature. Some are passenger mutations or neutral. And so this really speaks to the idea of we're building an atlas, we're building a catalog, a compendia of all the mutations and alterations that are identified in human cancer. 50 different cancers, 500 to 1,000 tumors per cancer. Comprehensively annotate on every level. But that, again, just gives us lists. So I speak to the idea of building the functional cancer genome atlas. Functional in the sense that how can we best functionalize all of these data that we're getting from TCGA to inform on those targets, those mutations that are active. So we can inform on genetic sufficiency. So this is more gain of function, like a study I just showed you in melanoma. So some of those mutations were sufficient enough to make a cancer in the mouse. Some of them weren't. So is the target a driver of disease pathogenesis? Can you take a benign melanocyte and make a melanoma just by simply overexpressing one of these genes or not? Inform on genetic dependency. Are tumor cells dependent on the expression of this target? Again, I'll talk a little bit more about the technologies that allow us to do that. Is the target activity or expression required? This is the level of validation which helps us prioritize whether a target is good or worth investing in for drug development and discovery. And lastly, mechanisms of resistance. We can engineer cells. We can do amazing things to try to, and I'll show you one study by a colleague of ours. We can engineer them with a panel of genes in an arrayed format, one gene at a time, 500 genes, 1,000 genes, 15,000 genes. Now, which ones, when expressed, confer resistance to a particular drug? So we have those technologies available to us. So this is really the path towards functional interrogation of cancer genomes. It starts with a target cell. So this could be a generic cancer cell. You can go to companies that will sell you what we call generic cancer cells. These are cell lines that have been grown on plastic for 40, 50 years. You can engineer cell lines, primary engineered cell lines. I'll show you an example of how we did this a little bit later on in the talk. You have a number of different technologies, or libraries. So these are open reading frames. So in genetics, you see the sequence of a gene. You have a promoter, you have a start codon, excuse me, and then you have your genetic sequence. So it's the central dogma. Have you read that in your textbook, where you go from DNA to RNA to protein, right? And so what we have here is a collection of all the DNAs of as many genes that one could clone. Not the promoter, just the sequence that creates RNA. And then you also have this technology of loss of function. This was the reason for the Nobel Prize a number of years ago. Andrew Fire and Craig Mello, showing that you can build short hairpin RNAs. That if you have a single-stranded RNA that's about to be transcribed, translated into protein, that if it is bound or shown to be double-stranded RNA, it looks like a foreign pathogen. And so what happens is the cell activates a response that chops that RNA up. So, building on something that is unique to our own cells, we took advantage of that. So now you can express these hairpins that will bind to specific RNA structures and cause your cells to chop them up. So you can knock down expression. You can do this stably. You can do it transiently. You can do it in inducible constructs. And so there are a number of ways that we can manipulate a cell to inform, one, can we make it a cancer using engineered cell lines? Or two, can we inhibit the growth of cancer by inhibiting the expression of certain genes? So what do we have available to us? In our own collection, we have a collection of 18,000, yes, yep.
So how that approach compares with our RNAi? That's more
The exact same thing. The exact same thing. So RNAi is the process, RNA interference. But classic RNA is using oligos, a pool of oligo in which there are 20 mers. You transfect them into cells, they bind to RNA, and that RNA gets chopped up. But with small hairpin RNAs, these are a little bit more stable. These you can engineer into expression vectors. And so this hairpin structure, again, within cells, is recognized as double-stranded RNA. And so what happens, a protein called dicer comes in, takes this hairpin, chops it up, and what it does is create all of the oligos that you speak of. So it's just one additional step on how we've evolved the technology. So the hairpin becomes the oligo. But you can manipulate it a little bit differently. You can make stable knockdown rather than oligos that aren't necessarily, they don't recombine into the genomic DNA. They're not carried from progeny as cells replicate. So you have a finite amount of time that you can study RNAi. But with hairpins, they stably intercalate into your DNA. And so these can pass for several passes. But our collection of ORF libraries, we have 18,000 ORFs. As of next week, we'll have 34,000 ORFs. We're building our second collection. 15,000 unique genes. Anybody know how many genes there are in the human genome? How many proteins, I should say? How many proteins? About 20,000. All right, about 20,000 genes that are expressed. Anybody know how many base pairs there are on the human genome? Individual bases. About 3 billion. And so of the 20,000 proteins that are expressed from the human genome, we have the ability to express about 15,000 of them. We have built libraries both in pooled and arrayed formats. Pooled meaning we can build these lentiviral constructs, each one expressing a different gene, and we can pool them all together. So we can have pools of hundreds of genes, pools of 1,000 genes. Sprinkle that on cells and see what happens. It's incredible. In contrast, through the efforts of Bill Hahn and David Root at the RNAi Consortium, people like Steve Elledge and Greg Hannon, we've built genome-scale shRNA libraries. We have built hairpins that will knock down every gene in the genome. Again, both in pooled and arrayed format. And now, as the technology has evolved, we can do a number of different things. Again, we can express hairpins in an inducible format, where you can engineer cells to express the hairpin, or to intercalate the hairpin into its DNA, but you can determine when the hairpin will be expressed, right? And this is all based on promoters, different promoters. So you can say, I want to knock down EGFR in a lung cancer cell line, but I'm going to do it in vivo after a tumor has been established. And so you engineer the cell line with the hairpin, and then by adding a inducer of that promoter, you can say, all right, on day 10 days from now, I am going to activate that hairpin. And so these again, just like sequencing technology, genetic technologies continue to evolve. And so we have all of these types of activities available to us. And so what it allows us to do is to study a number of different cancer phenotypes. We can study the effect of overexpressing or knocking down a particular gene product in the process of in vivo tumorigenicity, in vivo metastasis, anchorage-independent growth, again, a hallmark of cancer, ability to grow without, the binding to a basement membrane. Invasion or migration. This is a poor picture. I'm not much of a graphic artist, but what this is trying to show that if we put cells in the chamber on the top, we can force them to migrate to the bottom. Proliferation or apoptosis, right? If I overexpress 10 genes, will any of them make cells grow faster than the cell alone? Drug resistance. Again, I'm going to show you a number of examples of how we've used these technologies to inform on these cancer phenotypes. And this helps us take a list of genes, 1,000 genes, 15,000 genes that we can express. Which ones are critically important for these cancer-related phenotypes? And so the first story I'll tell you is, again, the poorest picture on the slide, is a study that a colleague of mine, Ken Scott, and I did when we were postdocs in Boston. We were fellows. This technology was starting to become available. Just like anybody, we thought we could do it better and faster, and we could, change the technology for the better. But it actually worked out. I was surprised. So what we did, in the study of metastasis, you have a primary tumor. This tumor proliferates. It signals so that blood vessels will form. Just like anything else, cells need oxygen, right? Cells need oxygen to create energy. And so this is the whole idea of, a blood supply. So cancer tumors send out a signal to say, okay, we need a blood vessel here. And so the blood vessels will seed. And then through these blood vessels, you have detachment or invasion, where a cancer cell will invade through the basement membrane into the blood vessel, which is leaky. And then this is the process where it enters circulation and is now able to enter the lymphatic system, go to the lymph nodes, metastasize to the liver, the brain, or the lung. So this is the process that's absolutely critical. It gets transported from our own circulatory system through the heart, through the lungs, adherence to a vessel wall, and then it metastasizes. It exits the blood cells, can seed because of anchorage-independent growth, can seed within the lung, and then once there, can develop into a tumor. This is what is informing on mortality more than anything. It's the metastatic disease. Oh, I thought I had a question. I'm sorry. And so Ken Scott had this idea. Could we set up a system in which we could measure the effect of overexpressing a number of genes on invasion using a cell line that does not invade? How could we confer this phenotype? And so this really started with our own genomic sort of technologies back then, where we took, and I'll talk about this later on how we use mouse models of cancer to help us inform on human cancer pathogenesis. But in this case, we took 2 melanoma mouse models, right? Mice can be engineered. Have you talked about mice in your course? And did you have that this week? Yeah. Did someone present this? Perfect. So as you know, you can genetically engineer mice to express certain oncogenes or tumor suppressor genes. And so we had 2 melanoma mouse models. One that was driven by NRAS, RAS, the other undruggable, the elephant in the room, commonly mutated, 90% of pancreas cancers, lung cancer, colon cancer. If you can drug this, incredible impact. So far undruggable. One driven by RAS, which was non-metastatic. It would only form primary melanomas on its back. Then we compared it to a different mouse model that expressed MET. This is an RTK, known to drive invasion and metastasis. And so we had 2 different mice that we could compare. One that formed metastatic tumors and one that didn't. So then we took those tumors and we annotated them genomically. First, we looked at expression differences. So if you take one tumor that's a MET, one tumor that's a primary, they're both primaries. One can metastasize and one cannot. So something in this cell is different from this cell, a tumor. We found almost 1,600 genes were differentially regulated. Some were up in the non-metastatic, some were down in the metastatic, and vice versa. So we had a starting point of 1,600 genes that were different. We then integrated this with genomic information that we had from human melanoma. Again, primary melanoma versus metastatic melanoma, looking at CGH plots. And as you can see here, again, this is recurrence. These are chromosome numbers. Red going up means amplification. Green going down means deletion. And so from this analysis, we integrated both mouse and human data, and we came up with a list, what we call metastasis determinants, or a signature, where 295 upregulated genes had been identified in mouse and human, 65 downregulated genes. Again, mouse and human. So how do we inform on which of these 295 might be important? And so we built a functional genomic screen. So Ken, this was, again, really advancing the field, when Ken built this. So again, we compared RAS versus MET, human melanoma, the genomics. We came up with a list. We built them into lentivirus. 295 genes, packaged them all into lentivirus individually. We expressed them in a normal melanocyte, what we call primary melanocyte. I'll speak to this in a few slides about how we manipulate and engineer individual cell lines. But for the sake of this slide anyway, we immortalized it with telomerase to give it a infinite lifespan. We took away the tumor suppressive arms, P53 and RB. I know you talked about P53 yesterday. And then we, again, expressed BRAF V600E. I'll talk more in detail about these cells, but nonetheless, this is a cell line that does not invade. It has no invasion capabilities. We engineered these cells to express the 295 genes. We entered them into a 96-well invasion assay, very high throughput, to identify those genes that could confer an invasive potential. As shown here, GFP is more of our control. So this is background, what you see on the top. But you can see, especially clone number 10, when you overexpress these genes, you can confer invasion. So these cells will basically eat through a basement membrane and invade to a portion that has more mitogenic stimulation, more serum, more things that allow it to grow. So you can see here we have 12 validated hits that scored twofold or greater with respect to invasion. We went on to validate, through Ken's efforts, using in vivo, again, models. Let the mouse tell us which one is important. We took a gene called HOXA1. The mechanism of this has just recently been published last week. Took a HOXA1. If you inject them into the tail vein, right into the vein of the tail. So it goes directly to the lung, right? So here we're measuring lung seeding. You're measuring the ability of a gene, HOXA1, when expressed in a cell, that if we inject it into the tail, into the vein, the vein of the tail, will seed the lung. And that's what's shown here in this gross histopath picture here, where you have now seeded the lung. So these are mouse lungs, this is the heart, and you can see this tumor here. And this is shown up in histopath. One of the better pictures is this one. Here we're looking at distant mets from an orthotopic site. So we engineered melanocytes to express HOXA1. We engineered the cell lines to express green fluorescence protein so that we could track it within the body of or the organism, which is the mouse. Here is your control. When you look at the liver or lungs, you don't see any GFP. If you overexpress HOXA1, you see all of these cancer clones as shown by GFP staining. So again, we started with mouse and human data. We started with a list of 1,600 genes. We prioritized that down to 300. We ran a screen to prioritize that down to 20. And now we've validated several of these. One of them I'm showing you here, HOXA1. And so we found a gene that is overexpressed in metastatic melanoma that can confer this phenotype. again, that's the level of validation and prioritization for us to say, this is an important gene. If we started with 1600, how might we have guessed that this was the gene that would confer this invasive potential? More so, when you look at the signature that Ken built, the 20 genes, and you look at expression correlates using in silico platforms and online tools such as Oncomine, which measures the expression across different tumor types, you see that many of these, melanoma, glioma, breast cancer, pancreas cancer, lung cancer, a number of these are overexpressed in a number of these different cancers. And if you take the signature of the 5 top hits and you put them into a program for breast cancer, we can show that if you stratify patients based on the expression of these genes, it correlates with a poor prognosis. Exactly what we would expect from something that confers an invasive or metastatic potential. And so this is one example of how we've translated the human genome. We've identified a number of genes by simple expression analysis using 2 different mouse models, integrated that with human data, and informed on a number of genes that stratify patients for poor prognosis in breast cancer. When we think about other phenotypes, one is drug resistance. You know, is there a mechanism that we can use a lot of these tools to inform on how patients might relapse from certain drug treatments? This is a study done by Levi Garraway, a collaborator of ours, where he took a collection similar to the collection that we have of 600 human, this is not in your, sorry, 600 human kinases. And again, we can engineer libraries of the human kinome. We can look at G protein couple receptors. We can look at metabolic enzymes. We can look at epigenes. So what Levi had done in this study was to take 600 human kinases in an arrayed format, express them, engineer cells to express each of the 600 kinases, overexpress them, and then treat those cells with vemurafenib, or the BRAF inhibitor. And so what he found is the majority of these didn't do anything, right? The vast majority. You know, 550 of the kinases had no effect on resistance to BRAF inhibition. But then he found several. The control, constitutively active MEK, that's downstream of BRAF. So that's your positive control. But what he found is a number of these others, including MAP3K8, which is also known as COT1. He has gone on to show that COT1 copy number in cells that have amplified COT, on the bottom, they are resistant to vemurafenib, or Plex 4720. And so again, not to go into the details of the experiment, but more so the model, that we were able to take the entire kinome, engineer cells in vitro, treat them with a drug, and then identify a mechanism of resistance. So Levi has gone on with clinicians at Mass General Hospital to show that COT1 is expressed in melanoma tumors upon relapse from vemurafenib treatment. That's one of many ways to confer resistance. But then that also informs, again, on rational drug-drug combinations, where you could combine a COT1 inhibitor with that of a BRAF inhibitor. So again, functional genomics and translating the genome. But for us, this was the invasion screen and the resistance screen. These were two in vitro screens that, that we had been working on. We were more interested, based on the success of the invasion screen and the validation in the mouse, to do more work in vivo, right? Let the mouse tell us, let the organism tell us which genes are more important. And in fact, in a screen that we built, comparing kinases that were predicted driver mutants, we engineered melanocytes to express all of these kinases. We performed an anchorage-independent growth assay, where you take cells and you seed them in a semi-solid media, and then you ask which ones will actually grow, form a colony, versus those cells that have been injected under the skin in the subdermal compartment into a nude mouse. And what we found is that the hits are actually, you get a 50-50 overlap. between in vitro assays and in vivo assays. And this was more important for us with those hits that came out of the in vivo screen, because you control for the tumor microenvironment. This is not on a plastic dish or a semi-solid media. This is an organism, right? So there's the interaction between the host microenvironment and the tumor. It also uncovers non-cell autonomous hits. Those hits that are driven by the microenvironment, driven by the host to be oncogenic. And so we built a system that was really based on the work by Bill Hahn and Bob Weinberg of a number of years ago, where if you take a cell line, an embryonic kidney cell, you're going to express telomerase. And if you put in the viral oncoproteins like SV40, large T, and small t, which inactivate the tumor suppressors you talked about yesterday, P53 and RB, this is not, this might actually be in your thing. But if you add all these components into a primary cell line, you can force them to be oncogenic. They will form a tumor in a mouse. And so building on this strategy, we built a system in melanoma, in which we took a melanocyte, a human melanocyte, primary, genomically stable, does not form tumors, doesn't even grow past passage 10, immortalized it with HTERT, tried to avoid viral oncogenes, expressed the dominant negative form of P53, a constitutively active form of a kinase called CDK4, again, removing all of the tumor suppressor pathways. And then we expressed BRAF V600E. Again, 80% of benign nevi express BRAF, so we wanted to inform on what could we do to identify genes that could cooperate with BRAF. Again, rational drug-drug combinations. The system that we built is a context-specific screening platform. We have target cells that capture genetic context or definable subtypes. The library, what we call GEOIs, or genetic elements of interest. These are genes that are deregulated in human cancer. These could be enzyme classes like kinases I just showed you. They could be genes that are conferred through invasion or migration assays. And then the phenotype was going to be in vivo. How can we take a cell line that is a normal cell line, engineer it, inject it into a mouse, and form a tumor? I'm not going to go through this, just in the interest of time. But nonetheless, what we found is that when we took a focus driver kinase library, these are 110 wild-type kinases, we injected them in a pooled format, engineered them into human melanocytes, injected them into the mouse. As you show on the far right, we formed melanoma. Right? We went on to take that tumor out, analyze it genomically, perform PCR, identify the genes that were conferring that growth advantage. We identified the junk pathway. This was very surprising to us because the junk pathway has been shown to be pro-apoptotic in a number of tumor types. But in melanoma, in the context of BRAF mutation, junk is actually pro-tumorigenic. It's an oncogene. We've gone on to show, and I'm not going to go into too many of these details, we've gone on to show that we can transform human cells, human melanocytes, and mouse melanocytes. This is just tumor-free survival. So you inject 10 mice. 5 of them form tumors. Inject 10 mice. 10 of them form tumors. It's percent survival. So we can transform otherwise non-tumorigenic cells to form tumors. We can show using the technology like RNAi that if we inhibit the expression of JNK2, which is a potent kinase, we can inhibit the growth of human melanoma cell lines in culture. You have some cell lines that don't respond. This is just measuring colony formation. You knock down with 2 independent shRNAs, you inhibit growth. We show that the response is associated with activation status of that signaling pathway. So you're integrating with functional proteomics. So this was the killer experiment, right? This is what we call the killer experiment, the go, no-go decision with respect to a drug development endpoint. Tumor initiation, using the inducible hairpins that we talked about, that I can determine when these hairpins will be expressed. So we studied first tumor initiation. You turn on the hairpin. 2 days later, you inject them into the mouse. If you do that, these tumors don't grow. So this is a doxycycline inducible system. If we add doxycycline to the culture media, the hairpin is expressed. And so what happens here is red is the tumor alone, is the cell line alone. And as you can see, these tumors grow pretty rapidly. If we knock down the expression of JNK2, they don't grow. So we know that it's required for tumor initiation. But again, we're not treating patients for tumors that haven't grown or haven't established in the patient, right? So we're trying to inform on targets that if we extinguish that activity in tumors that are established, will those tumors stop growing or grow slower? And so that's tumor maintenance experiment, where you inject the cells, you let the tumors establish, and then you turn on the hairpin. You knock down that target, and then you see what the consequence is. So as shown here, these tumors are exactly the same size at day 10. We randomly separated the two pools. One group got placebo, or sham, or corn oil, whatever the hell we used in that particular experiment. And the other group got doxycycline to activate the hairpin, to knock down our target. And what you see here, even though we're only knocking down protein by about 60%, we see a 50% tumor growth inhibition. Almost stasis. Near the end of this experiment, these tumors are no longer growing. To suggest that if we inhibit JNK2 in BRAF mutant melanoma patients, we could inhibit tumor growth. Now that's a far stretch from a single mouse experiment, but this is the data that would suggest to us this is preclinical proof of concept, genetic proof of concept. Now we can think about prioritizing this for drug discovery.
First thing we talk about the TED-off, the TED-on
Yep.
plasmids. This is one of that.
This is exactly the same. So doxycycline is an analog of tetracycline. It's cheaper. We save a little bit of money. But what this allows us to do is this, like I said, the killer experiment. If we saw no difference in this experiment, kill it. End the program. Publish it. Tell people that it's, tell Big Pharma that it's not worth investing $1 billion in this target. But since we did see, and believe me, I have plenty of examples of those, where we don't see any effect, right? But this is the type of data that gives you that confidence that maybe this is actually required for tumor growth. So these are the type of experiments that we do. So this is the TED-off system. And that, again, the evolution of that technology allows us to do this type of experiment with genetics. I'm not going to go into too much detail. We've shown that in human tumors, we have activation of the pathway. But this led to the clinical path hypothesis. This is a very simplified, this is what signaling pathways probably look like in your textbook, right? Straight up and down, right? It's so easy. And so we took advantage of that, right? So here you have the signaling pathways that are activated in melanoma. You have the MAPK pathway. It's activated by RAF, MEK, and ERK. You have the PI3 kinase pathway. PI3 kinase, AKT, mTOR. Both pathways cooperate to activate or stimulate growth. Then you have the JNK pathway, which again, in some contexts, is pro-apoptotic. In melanoma, it's pro-tumorigenic. This drives the activation of a transcription factor called AP1. Changes the whole expression of a cancer cell line. And so our clinical path hypothesis was to combine a RAF inhibitor, like vemurafenib, with that of a JNK inhibitor. This program was initiated with, in collaboration with Merck Research Labs. We worked for about 2 years to identify a potent selective inhibitor against JNK2. The studies are still ongoing. But again, through functional genomics, we've been able to inform on a number of these targets non-obvious clinical path hypotheses that are context dependent. And so that's really the basis of a lot of the studies that we do. I'm not going to go through this much, but just simply to say that we've, based on the power of the melanoma and the success of that approach, we've built systems in a number of disease indications. Breast, GBM, ovarian, melanoma, lung. So we're continuing going through this process where we can look at, for instance, in GBM, we can, through genetic engineering of these cell lines, we can build cell lines that mirror all 4 of the subtypes, genetic subtypes in glioblastoma. So through the same approach, I could inform on a target specifically for the proneural subtype, which there is no drug. So again, the power of this approach. We could look at genetic alterations. This is, again, the messy CGH profile of ovarian cancer. I have primary ovarian epithelial cells that express MYC or PI3 kinase. Two patient populations that would be, would love a new drug to treat as standard of care, in addition to platinum. Or pathway knowledge. What pathways are activated in what cancer? So using functional genomics, we could inform on a number of these approaches. This has spurred our activity in this US-driven CTD squared network. So this is the Cancer Target Discovery and Development Network. As you see in the small box in South Texas is the University of Texas MD Anderson Cancer Center. This is a program that myself, Ken Scott, who we are now working together again after several years apart after our postdoc, Gordon Mills and Linda Chin. We are building platforms that we could inform on novel targets in a context-specific manner. This is the platform. Using sophisticated computational modeling, go through each of these genome data sets to inform on those genes that are likely more functional, like the invasion screen. We compared 2 different mouse models. We integrated that with human data. Now we have a number of genes that confer an invasive potential. Here we're going to do it on all genes, integrated data from TCGA. We're going to engineer these alleles with putative somatic mutations. We can engineer by simple PCR methods somatic mutations through very common mutagenic protocols. I'll put the wrong emphasis on the wrong syllable there. You can assess the putative driver mutations in vitro studies. And then we'll put them through our catalog of target cells for in vivo analysis. This is an incredibly complicated slide. But I put it up there because Ken Scott put the time in to build it. Right? This is how we build our functional genomic platforms. So this is essentially saying we're going to data mine in AIM-1. We're going to query our ORF collection, which is now about 35,000 different genes. We're going to put them into our cloning pipeline. We're going to batch the libraries. We're going to perform our mutagenesis. And then we're going to enter them into our screens. This is all fully automated approaches. So we use robots to do this. I'm way too tired to do this by ourselves. But with robots and automation, we can scale this up. Very scalable. Has anybody performed site-directed mutagenesis? Has anybody performed PCR? PCR? It's essentially PCR. You just use a different primer. You use a primer that has that mutation that you want to engineer. So by standard PCR, you're basically elongating from the end of that primer. So what happens is you basically replicate the entire plasmid. In the end, you have your cDNA with your anticipated mutation. But now we have computers to tell us what primers we should use. We have automation to help us prepare the DNA. Automation to set up the PCR. PCR machines are PCR machines, right? And so we can do this in very high throughput. So what we aim to do is to build 3,000 new mutant alleles within 1 year. And so it's 3,000 different PCR reactions, 3,000 different sequencing reactions, and so on. 3,000 is, we want it to be more conservative. We'll probably make 5,000. But that tells you the type, again, with respect to translating the genome, we now have the ability to engineer the same mutations that are being identified in patients' tumors. And so we can determine which ones are more functional than others.
That system works around the clock, non-stop?
It doesn't work around the clock. But it does work during working hours. So it's functional about 12 hours a day. Yeah. Yep. And so this is the idea. That we would use the computer, use in silico platforms to tell us which mutations, which genes are important. We would build them into our constructs, as shown here. Make all the mutations, express all the wild types. So of course, when you're trying to inform on the activity of a mutant allele, you also want to compare it to the wild type. So we're talking an order of about 10,000 different alleles that we're going to test. We can build all the virus. This is all antivirus based, high throughput. We're going to test them in viability assays. determine which ones make a cell grow faster, which ones become resistant to an Informa drug library, and then lastly, pass them through our context-specific screens. So this is the idea that through the CTD network, which I've shown you here, there are a number of different activities ongoing. California, up in Boston, New York, Georgia, with the singular goal of making this all public. Together as a group, we will functionalize the cancer genome. Whatever we'll find, we'll put out into the public databases so that people like you, inquisitive minds, will take a look at that and say, hey, I want to work on that gene, or that looks cool. Or wow, this is, I want to go to grad school or teach or something. But collectively, as a group, the public, everybody studying oncology and genetics can help inform on the next generation of drug targets. And that's the goal of this program. How's everyone doing? Good? Yeah, that wasn't very positive. Can we finish up this last section, then we'll take a break? Do you mind? Sure? All right. So this is a section on genetic dependency, and I'll go through this pretty quick. So have you heard about synthetic lethality at all this week? So this is a classic yeast sort of phenotype, where you have two genes, gene A and gene B. When both of them are wild type, this is not in your thing. Definitely this section. So just relax, sit back. Don't worry about, this is not in your handout. So you have two genes. When they're wild type, the cell is viable. When you have a mutation in A, it's innocuous, neutral, still viable. You may have a mutation in B, still the same, perfectly viable. However, when you have mutations in A and B together, it's lethal. Synthetic lethality. That context, right? That's classical synthetic lethality. When thinking about genetic synthetic lethality, again, if we use hairpins, RNAi, if we inhibit the expression of gene B when A is wild type, no effect. But when A is mutant or lost and we inhibit B, it's lethal, right? These cells die. Pharmacological synthetic lethality, which we're most interested in. Again, if you inhibit gene B, protein B, in the context of wild type A, nothing happens. Viable. However, in the context of a mutation in A, if you inhibit B with a small molecule, the cells die. Right? So why is synthetic lethality so important? So one of the problems that we're facing with respect to treatment decisions in the clinic using cytotoxics. I spoke about this earlier. Cytotoxics target every cell in your body. Again, every cell that's replicating. That's why you see alopecia. You lose your hair, nausea, vomiting, lose weight, immunosuppression. Right? Can we develop smart drugs that only recognize or are effective in cancers? Right? That won't affect a normal cell. So this is the concept, one concept of doing that, and that's synthetic lethality. In most of the cell lines, the cells in your immune system, in your hair follicles, in your gut, they won't have mutations in gene A. So inhibiting gene B will do nothing. Your normal cells will be spared. But in the tumor cells that are driven by mutations in gene A, if we inhibit gene B now, we'll have tumor-specific responses. The best example of this, which I'm sure you'll hear about tomorrow by Karen Liu, is the synthetic lethality or targeting PARP in tumors with germline BRCA mutations. Who's heard about BRCA? Okay, BRCA 1 and 2, mutated in breast cancer, heritable breast and ovarian cancer. Right? So in normal cells, BRCA1 is wild type, you inhibit PARP, nothing happens. That's shown down here. Using a PARP inhibitor, wild type cells are shown in blue. They don't respond. But in patients with BRCA1 or 2 mutations, breast cancer and ovarian cancer, mutant BRCA1, you inhibit PARP, it's lethal, as shown here in red. Right? So this led to this work from Alan Ashworth and others, but this led to the explosion in the field of synthetic lethality, and more specifically, the development of PARP inhibitors. because there you have a biomarker. BRCA1 mutations, these patients would respond to a PARP inhibitor. Now, how does this work? This is more DNA repair. I don't want to get into too much detail, but PARP is required for single-strand base repair. Have you got into that yet in genetics, DNA repair? Yeah? You guys like it? Enjoyed that section? So PARP is required for single base excision repair. Single bases, UV type stuff. Now, in the context of PARP inhibition, normal cells will repair that DNA using homologous recombination, a separate pathway. That's driven by BRCA1. However, in BRCA1 mutant cell lines that are deficient in this other repair pathway, if you inhibit PARP, there's no way to repair that DNA. It becomes genomically unstable. The cells end up dying, right? And so again, synthetic lethality. Can we select patients that would respond to one inhibitor versus the other based on their genetics? So PARP is a classic example. This says now, like I said, there's been an explode. Yes?
So that only works, can you put this?
Yeah.
That would only work, synthetic lethality would then only work if the first gene is dominant.
Right. Absolutely.
So that would work for...
Well, no, not necessarily dominant, but redundant.
So it would only work for tumor suppressors, not oncogenes.
No, absolutely. No, it works for oncogenes as well. So it's redundant functions.
Okay.
Or, as you say, one example of dominant functions, as you say, I think it's a great question. In the next couple of slides, we'll talk about how people are trying to inform on synthetic lethality with respect to oncogenic mutations like KRAS mutations or MYC. These are very different from normal cells. KRAS would drive a completely different transcriptomic profile or program. And so that would be synthetic lethality in the context of KRAS. That would be dominant. But with respect to PARP and BRCA1, it's redundant functions. You need to repair DNA. If you have two pathways, if you inhibit one of them, you're fine. If you inhibit both of them, it's lethal. But this type of activity has led to this real explosion, led mostly by Bill Hahn, Steve Elledge, and others, where they can take, again, genome scale shRNA libraries, where they take your reference condition, your test condition, you engineer cell lines with genome scale hairpins, talking about 200,000 individual hairpins. You grow them for a number of passages, and then you, by deep sequencing, you can determine which hairpins are still expressed. If the hairpin is not expressed, that particular cell had died. It was lethal. So again, you can see enriched, no change, or depleted, right? And so this type of activity has really informed on a number of different targets that are synthetic lethal with specific genomic alterations, like KRAS. This is Project Achilles, a paper recently published by Tony Chung. Jesse Boehm was a good friend of mine from Bill Hahn's lab at the Broad Institute, where they took 100 cancer cell lines, did the same assay, knocked down every gene in the genome, and then determined which genes were lethal when knocked down. And so as you can see here, all the cell lines are across the top. Normalized fold enrichment. These are all individual hairpins. So then they can say gene X, which might be TBK1, is only lethal in the context of KRAS mutation. If you have wild-type KRAS and you knock down TBK1, nothing happens. And so this type of approach across a number of different cancer types is starting to inform, again, on novel targets. A completely separate but yet equally informative functional genomic platform. In the end, for us, it's to compare the response in cancer cell lines to the response of tumor models to the clinic, right? We'll talk about this in the next hour, but this is the idea that has led to using, you don't necessarily have to use genetic means of manipulation. You don't have to use the 34,000 ORFs that we have. You don't have to use the 200,000 hairpins. You don't have to manipulate the expression of genes, but yet you can use drugs. You can use chemical tools or probes that are selective for one protein versus another, one enzymatic function versus another. And so two papers that were published in Nature this year, one by a group at the Broad led by Jordi Bartina, Levi Garraway in collaboration with Novartis. They're building the Cancer Cell Line Encyclopedia. This is an incredible effort in which they will compare these studies by Bill Hahn with the Project Achilles and then test all of these 500 cell lines with a number of different drugs to determine which cell lines will respond and what are the genetic associations of response. And I'll go into that in a little bit with respect to context. There was also an effort that was started at the Sanger Institute when Andy Futrell was still there. So they're doing a similar study. Again, systematic identification of genomic markers of drug sensitivity. This is all in vitro, taking a number of different cell lines, sprinkling on some drugs. Do the cells die? Do the cells smile? Do the cells grow better? Which happens sometimes. So, and then taking a step back and saying, okay, these 10 cells responded. You have 100 lung cancer cell lines. These 10 lung cancer cell lines responded. What makes these 10 different from these 90? Right? That's biomarker driven. Those are the type of, and this is functional pharmacogenomics. I don't know. That's what I would call it. Right? You're using chemical tools, integrating that with genomic tools, genetic tools. It's all making smart drugs, informing on why one patient will respond and another won't. And this is, let's see, the last few slides, and then we'll take a break. But I talked about, did I tell you that a few slides ago? Just a few more slides. This is an interesting section. This is an interesting section, more than anything else. And this is not in your handout, ironically. The most important part of the presentation, but we'll get it to you. I've spoken about this several times this morning about context, right? Context is so important. And I'm going to show you a number of examples over the next couple of slides on how important context is. Genetic context. BRAF versus NRAS melanoma. I'll show you a slide on this. If you take a BRAF inhibitor, vemurafenib, and you treat BRAF mutant melanoma patients, you see incredible response. If you treat NRAS mutant melanoma patients, you potentiate the disease. You make it worse. EGFR versus KRAS in non-small cell lung cancer. I'll show you a slide about this context. Notch. What genomic analysis of cancer genomes has taught us. It's context specific. Notch in a T-cell leukemia, ALL. The mutations in Notch, which activates a transcriptional program, are oncogenic. They're pro-growth. However, in head and neck cancer, squamous cell cancers of the skin, it's a tumor suppressor, right? Same thing with junk. So Notch is pro-growth and pro-death, depending on the organ in which it's mutated. So clearly you don't want to give somebody who has head and neck cancer a Notch inhibitor. Again, it would make it worse. They did this with Alzheimer's. They were treating with a gamma secretase inhibitor. They started to get squamous cancers on the skin, all due to inhibition of a tumor suppressor. Cellular context, we talked about this a little bit. EGFR inhibition in non-small cell lung versus GBM. I'll show you a few slides of this. Again, a brain cancer is different from a breast cancer, is different from a colon cancer. BRAF inhibition in melanoma versus CRC. We'll talk about this. Microenvironmental context. I introduced the stromal barrier in pancreas cancer. How can you treat a tumor when you can't expose it to drug? These are the challenges that we face. With genetic context, it's very important to understand that, like your textbooks show you, signaling is not linear. And this is just an example reviewed by Larry Kwong and Linda Chin. If you have BRAF mutant melanoma, you are driving activity through ERK. If you treat with a BRAF inhibitor, you inhibit ERK phosphorylation. Those cells die. Those patients respond. They relapse eventually, but they respond. If you do this in NRAS mutant melanoma, you potentiate the disease. You activate a different RAF, CRAF, a brother, a neighbor, friend, that can maintain this activity. But it's much more powerful. These tumors grow much greater than they would be under treatment with standard of care. So context is important. Biomarker driven. KRAS versus EGFR. Right? Lung cancer, colon cancer. You have populations of both cancers with both genotypes. EGFR amplification or mutation, KRAS mutation, right? If you treat KRAS mutant, let's just go down to the bottom. 25 patients with KRAS mutant lung. You treat with an EGFR inhibitor. 2 out of 25 respond. 8%, right? Again, same thing, KRAS mutation negative, right? So these are wild-type KRAS patients, EGFR mutant, 26% respond. Cetuximab, another EGFR inhibitor, it's a large molecule, antibody. Colorectal cancer, if you have mutant KRAS, 0 out of 36 patients responded, 0%. If you're a wild type for KRAS, 44 patients responded, 44% of patients responded. So what genomics has told us, context is important. There are negative, not only are there positive predictors like the V600E mutation of BRAF, but there are negative predictors of response. You do not want to treat a KRAS mutant patient with an EGFR inhibitor. They're not going to respond, it's a waste of time. Treat him with something else that'll be more effective. We haven't known this. We haven't known this before. So these are the type of studies, the functional genomics, pharmacogenomics, these type of studies where we're using genetics and drugs, we can inform on what patients are likely to respond and which aren't. Over the next 10 years, you're going to see the whole clinical trial setup completely revolutionized, where we're not necessarily going to be taking all comers and saying everybody in this room is going to get drug number 1. We're going to separate you into groups by analyzing your genome, right? Adaptive learning. We're now utilizing, Linda Chin and Andy Futrell are starting a program at MD Anderson using artificial intelligence. Taking 1,000 patients with acute myeloblastic leukemia, AML. They're analyzing their genomes. They're putting it into a computer. Anybody know of Watson, the computer that won Jeopardy? So it was the supercomputer. It beat all the Jeopardy champions or something. Smartest thing on the planet. So what we're doing is we're dumping all of that computer in what we're calling the big data network. Everybody's tumor into Watson and having the computer tell the clinician, this patient should get what treatment, right? So artificial intelligence, genomics, and how that's really revolutionizing the field of personalized medicine. Very cool. This is the EGFR yes?
How far is that from reality?
Just started. Just started. So the pilot study will be done probably by the end of this year. But yeah, you can do this in silico. This is a great example, KRAS versus EGFR, this best experiment. But what we can take is a retrospective study, is to say, all right, these people who did not respond had this genetic association. But instead of doing it on one gene at a time, KRAS versus EGFR, we can do the entire profile, whole genome, and say, these are the patients that we know are going to respond. These are the patients you don't want to even invite them to the hospital yet, because we don't have a drug, right? So over the next year, we should see the power of the supercomputer and how this will do. Remains to be seen whether clinicians will listen. But I think it's an important step. This is an example of EGFR. Works in lung cancer. Works great in lung cancer. But in brain cancer, this is a CT scan, right? Here's a patient with a glioblastoma, the bottom left, right? 2 months on an EGFR inhibitor. Did the tumor get smaller? No. Doubled, 3 times in size. Tripled, doubled, tripled. Look, right? So context is important. EGFR inhibitor is not going to work in brain cancer. And we know the reason. This is a good friend of mine, Jane Stommel. We were postdocs together. She was working on this project. What happens when you inhibit EGFR? This is, again, a very simplified view of the membrane of a cell. We have receptor tyrosine kinases. They accept stimulation through ligand binding from the external environment. They trigger signaling downstream. If you inhibit EGFR, you just activate other RTKs. But like the BRAF and NRAS and melanoma, you make it worse. These tumor cells grow much more, much faster, much more aggressive. So context is critically important. Colon versus melanoma. Vemurafenib works in melanoma, right? Works great. 60% response rate. Same mutation, BRAF V600E, in about 5% of colorectal cancers. It's the driver of that disease. One would expect that if you treat BRAF mutant colorectal cancer with a BRAF inhibitor, you should see response, correct? You do not. It's incredible, right? You don't see response. And this is just to show, again, by Jeff Engelman at MGH. These are colon cancer cells and melanoma cells. This is looking at a downstream biomarker of response. We know in melanoma you have to inhibit phospho-ERK by about 80% to see response. We can't achieve that in colorectal cancer. We can achieve it for about 24 hours. That's about it. And the reason for this, again, is that you're activating EGFR. So when I showed you that spaghetti picture, where there's mutations everywhere, this is one mechanism of resistance. Again, you activate this pathway. ERK is through the roof, so you have to combine. You do a BRAF inhibitor plus an EGFR inhibitor. And this is preclinical proof of concept. This is in a mouse. Control does nothing. This is change in tumor volume. Erlotinib is an EGFR inhibitor. Very little. Not going to see much. Not stasis, not regression. Vemurafenib, very little. When you combine the two, rational drug-drug combinations, great efficacy. So context is important. So in the end, the conclusion of this first part, we have the tools. The cancer genome is informing on gene lists. We have genetics. We can overexpress genes of interest. We can knock them down. We have drugs. We have chemical probes, an armamentarium of sorts to manipulate genes or activities within a cell. We have to inform on context. Again, the most important thing, what patients are likely to benefit. We can study a number of different phenotypes, invasion, tumor growth, resistance. And functional genomics, translating this genome, will inform on context-specific targets. Will provide that clinical path hypothesis for the next set of clinical trials. And again, coming back to this, it's not the target. We've identified some great targets. It's not the drug. You need the drug, but that's not the end. It's the patient. We need to inform on the correct patients. All right? And we don't have to talk about this. All right, why don't we take a break? Thanks for your attention. Do you have a question? Oh, yeah, well, hold on 1 second. Sorry.
Okay, sorry. So what about, talking about pharmaceuticals, we spoke about small molecules.
Yep.
But where do protein molecules and protein type medicines come into play? Like engineering proteins, is that something that would be viable for the future?
Delivery is what's tough. That's the problem. Small molecules, they become bioavailable. You can take them as a pill. When they're not, you have to take them through injections. Large molecules like antibodies through IV drugs. But others, you know, there has been an idea of using RNAi in clinic. But it's delivery, which is the problem. And so with, you know, protein-based therapies, that's still an issue. Good. So how long do you guys usually take a break? And please don't tell me an hour. How long? How about 10? 10 work? So we'll come back, or how about this? At 10 past, we'll start again. So that's like 8 minutes. 8.5. So they serve lunch at 12 o'clock? Is that the idea? So we'll have, we got about 45 minutes, so we'll have, we'll go ahead and get started and then let the others trickle in. But, you know, in the first, you know, can't believe it was 2 hours, but the first 2 hours of, I hate to call it a lecture, but presentation was more on, you know, the complexities of cancer and how we're utilizing genomics and functional genomics to inform on patient selection. What I wanted to do in the next half, and I'm not quite sure we're going to get through all these slides, so I'll go through some of them pretty quickly. Some of them you have, some of them you don't, is to give you an introduction to introduce the Institute for Applied Cancer Science and give you a little bit of rationale on why we started and why we built the IACS, as we call it, and how we're taking a different approach than, say, big pharma or biotech with respect to drug development. And so we built this in September of 2011 at the MD Anderson Cancer Center. Obviously, there's a number of different advantages of working at MD Anderson versus any other. It's the #1 cancer hospital in the world. They see approximately 35,000 new cancer patients a year. Steady state, they're treating upwards of 100,000 patients any one day. And so for us, it gave us a unique opportunity to really build this sort of drug discovery engine. And it's really based on this premise here, where, you know, traditionally, drug discovery started in academia like many of you, through target discovery in biology. We would identify the target, we would publish it, say this target is important in colorectal cancer. Then biotech or pharma, big pharma, would then do the drug discovery and development. They would do the chemistry. They would develop that chemistry. And then they would go into pharma and back into academia for commercialization and clinical trials. But as you can see here, the process is way too long. It's inefficient, incredibly expensive, over $1 billion. I think we're underestimating this. But more so, through this 10 years, $1 billion, we're only seeing about 5% success rate in the clinic. And again, we believe this is conceptual. There's a knowledge gap. We don't know enough about the biology or genetics to inform on clinical development. We don't know who the patients are that should be treated. And that's really the goal. And so what we see here is that this is the typical approach. Basic research, novel target idea done in academia. Industry builds, performs a little bit of validation and drug discovery, builds a chemical probe, and then comes back to academia into our cancer centers to run our clinical trials, right? This is an academic bias, as I'll show you, and this should be shocking to you, that over 2/3 of academic publications can't be reproduced. This is not just because they're being fabricated, but this is because the models and the systems that we're using in our labs aren't necessarily predictive of the greater community. So 67% of academic publications can't be reproduced. So again, that's could potentially be contributing to the 95% failure rate in the clinic. The biology just isn't true. But this is shrinking. This drug development in industry, in big pharma and biotech. They don't have necessarily the preclinical models. When you work in big pharma and you want to license in a mouse model or a cell line, it may take a year to get through legal, get through processes to sign the appropriate MTAs. So drug development is shrinking in industry. And in academia, we need more innovative clinical approaches. And so what we wanted to do was build a system or build a program that bridged the gap between academics and industry. That we would build a unique construct that was goal-oriented, milestone-driven culture. We're not focused on publications. Will we publish our work? Absolutely. But will our jobs depend on us publishing data? Absolutely not. Our funding comes through investment from the state of Texas, philanthropic donations, grants. But it's not necessarily, our salaries are not supported through the endless cycle of publications and grant renewals. We have a professional industry seasoned staff. As you'll see, the IACS has over 200 years of pharmaceutical experience. We have members of our leadership team that have brought drugs from the initial steps all the way through clinical development. Genomics and computational biology. I spent 2 hours this morning telling you how important this was. You know, we have built a platform at MD Anderson and IACS with world-class genomics. Deep cancer biology and model systems. I'll show you, we're using both human and mouse. But performing the deep biology to inform on mechanism of action to say, if you have an inhibitor against protein Y, we can predict which patients will respond to that. And then the strong molecular pathology and translational medicine. This, again, being within the walls of MD Anderson makes this a lot easier. We were established in September of 2011. We started working in the lab about January of last year. You can see the people that we've recruited. Many of the leadership team came from the Belfer Institute, which is at Dana-Farber and Harvard Medical School. We've recruited people from all the major pharmaceutical and biotech companies. We have 57 full-time equivalents or employees. That's actually outdated. We're now at about 70. We've got about 5 more. We'll be about 75 people. Greater than 200 years of drug discovery experience within the team. And we started with an upfront investment of $75 million. So people are confident that our approaches to change the way we develop drugs and provide that clinical path for clinical success are worth the investment. We have 3 areas of focus. We have 1, the validation of novel targets, new target screens, functional genomics, as I spoke about this morning. We're running all of these platforms within IACS and the Department of Genomic Medicine. We build compounds, small molecules or biologics, antibody-based therapies, biotherapeutics. Not necessarily just tool compounds, but these are clinically developed and ready compounds. And we don't move on to the next step, as you'll see in a few slides, until we have preclinical proof of concept. So this is not, when we talk about the right target for the right drug, they almost seem next to each other. But the process of developing the right drug is an incredibly lengthy and expensive process. That's why we want to do it right from the beginning. Lastly, poor font, I would say, is informing on activity of a specific inhibitor in a defined genetic context, vemurafenib and BRAF mutant melanoma. So this is the process of developing a drug, right? So we read in the textbooks, Gleevec, we talk about erlotinib, Iressa, vemurafenib, crizotinib, but this is the process that's undertaken to develop a new drug. Not simply in oncology, but other disease indications. You have target ID and invalidation, lead ID, which we will go into. Lead ID is identifying a chemical probe. Lead optimization, optimizing the properties of that so it would be safe in patients, understanding what it does to the body. Early development, preclinical development. Can you show that it's active in a particular patient population? And then lastly, late development for clinical trials. So in target ID and target validation, I'll show you, it's building on some of the platforms that I discussed earlier. Functional genomic screens, I showed you a few examples, one being melanoma. We have now a functional genomics platform, both gain of function and loss of function, and a number of different indications. Multiple myeloma, a bloodborne disease. GBM, triple negative breast cancer. Melanoma, pancreas cancer. Prostate cancer, colorectal and ovarian cancer. Comprehensive, but not all of them. Again, we're informing, using functional genomics, we're utilizing the genomic knowledge that we're getting from all of the platforms we discussed earlier today. We have access to preclinical samples directly from the patient. This is a CT exam of a patient with a glioma. This tumor, as shown here by CT, has been resected. A piece of that tumor has been diced up on a plastic plate with a scalpel. Here we can generate spheres. A sphere is a tumor neurosphere. So we take these cell lines, we grow them on non-inherent plastic plates. They form a ball of tumor cells, right? These are two different patients. And what we can do is implant them directly back into the brain of a nude mouse. So we can rebuild human cancer in the mouse. And these are the systems that we're using, again, to perform our loss of function screens, perform our gain of function screens, trying to inform on how we would, using a statistically powered cohort of these patient-derived cells, how we can inform on new targets. Which targets are more patient relevant? Very much like Project Achilles, we're doing a lot of these pooled hairpin screens. This is just basically how we develop the pools. These are oligo-based, right? So we can package these into libraries. We can use both patient-derived cultures or mouse cultures and inform on which genes are required. If we knock down a gene and that cell dies, that's very important to us. Again, we use deep sequencing, imaging. But this also speaks to a lot of the preclinical models that we use, one of them being the mouse. So you talked about genetically engineering mouse models earlier in the week. We use those same models. What we've found, a lot of the pioneering work of Linda Chin and Ron DePino, Scott Lowe and others, that when comparing the genomes of mouse and human cancers, they're very much similar. If you look at some of these, the human is on the left. CGH profile that we spoke about earlier today. SCIP2 is amplified, MYC is amplified, RB, and KX3. Those are deleted. If you look at a prostate cancer in the mouse, you see the same genes, they're just on a different chromosome, right? So that's the power of utilizing mouse genetics, that you can compare your mouse models to that of human cancer. They faithfully recapitulate the disease. So that provides you, we don't have limitless human, patients willing to be in our experiments. Do you know what I mean? This provides us reagents, the mouse, that we can breed. We can determine when they will get cancer. We can determine what organ they will get cancer. So we have built, this is just a few. We have built an incredible cohort. Colorectal, GBM, non-small cell lung cancer, right? We use a number of genetic tools that you probably talked about on Monday. Different Cre deleter strains, inducible constructs, melanoma, pancreas cancer. Pancreas cancer, we have a full allelic series. Every common mutation that's observed in human pancreas cancer, we can express in the mouse. And they form pancreas cancer that is exactly what we see in humans. It's shown here. We express KRAS only in the pancreas, again, using tools that you discussed on Monday, with different tumor suppressor backgrounds, P53, INCARF, SMAD4, P10. These tumors form cancers, lethal cancers, very quickly. And so, again, cancer in humans is a disease of old age. Here in mice, we can develop a cohort of tumors that we can analyze in just a matter of a couple months. So utilizing the mouse models is incredibly important for us with respect to target ID. I don't want to go into too much detail. This is a novel pancreas model we recently published, work by Hao-Chung Ying, that we can express KRAS in a temporal fashion. We can turn it on and turn it off, right? Using the doxycycline approach that we talked about earlier, or TET approach. So here, 1 week on dox, KRAS is expressed, right? You take it off dox, within 24 hours it's no longer expressed. The tumors, this is MRI. This yellow ball here is the tumor. If we remove KRAS, the tumor shrinks. So this is a clear example of tumor maintenance. But KRAS, again, is part of the undruggable genome. But these are the type of models that can help inform. So we can do escaper type screens. And so when we think about utilizing both human and mouse, and we think about as an institute with X amount of dollars to invest in a clear milestone of developing drugs, how do we prioritize targets? How do we say this is a target we're willing to invest $10 million on in the next 2 years? And this really comes from this type of target assessment. We talked about this a little bit earlier. Biological function, is it relevant? Rationale, here we're using multi-omic data, cancer genomics included. Is it relevant? You know, is there evidence of deregulation in human cancers? Let's stay on pancreas cancer, right? We have in a full allelic series. We have patient-derived models. We have some of the best surgical oncologists and clinicians in the pancreas field at MD Anderson. Through genomics, if we identify a novel targeting pancreas cancer, we should be able to determine if patients with pancreas cancer are expressing that target. And that's step one. Feasibility of a screening funnel. We'll talk about this, but this goes into developing a drug. Preclinical models. Responder ID. Can we stratify patients based on this target? Predicted tolerability. If we know if we inhibit the activity of a protein, it's going to be toxic in humans, that's an absolute no-go, right? Toxicity is one of the issues that cause the failure and the failure rate in clinical trials. Tumor types, intellectual property, competition, go, no-go experiments. These are critical. Because we're not publication-based, we're developing drugs. We can design what we call killer experiments. We don't necessarily have to do everything. Our first set with respect to target validation is to say, you know, if we knock this down, do cancer cells care? If they don't, we kill it. We shelve the project. We stop working on it, pick up something else. Very different from an academic mindset where it's focused on publications and grant proposals. Here, we're focused on making drugs. So the highest priority targets are chosen through this approach. This is what I was telling you about. It's an academic bias. 2/3 of published data is not reproducible. This is not a statistic of my own. These were several biotech companies, pharmaceutical companies that actually did the study, invested the money into showing this. This was a Nature review from GSK. So because of this, we have very stringent in-house criteria with respect to validation. We're not going to go ahead and start drug development just because of a publication that we read 2 days ago. What we do is we start the initial validation. We try to reproduce the data, right? If we can reproduce it, great. It goes into our bin of targets. And then we assess those targets based on the criteria that I had just shown you. We use both genetic tools and chemical tools. There's pros and cons for both, right? Genetic tools, you know, when you're talking about inhibiting a target with a drug, you're not necessarily removing it from the genome. You're just transiently inhibiting it. So a genetic tool like RNAi, which extinguishes expression for a period of 3, 4, 5, 6 days, is very different from inhibiting a target for 4 hours. So chemical tools versus genetic tools. Many talk about tool compounds. These are chemical probes that may be not necessarily ready for the clinic. We have a very different definition of tool compound. For us, it has to be an incredibly potent and selective inhibitor. So as to say, whatever data we generate preclinically can be translated to the patient. This is really the idea of target biology and then drug discovery. So this should be included in your deck. And so you can read about this in a little bit later. We can talk about this over lunch. But the idea is simply to validate the target. Are tumor cells dependent on the target expression or activity? Clinical path validation. Is the target expressed in human cancers? Is it deregulated? Mechanistic study. What is the functional consequence of inhibiting that target? Right? So we know that if you inhibit BRAF, cells stop growing. Right? Cytotoxic. If you treat with a cytotoxic, it kills every cell. So those are the type of mechanistic studies that we need. Drug discovery, we're going to walk through this, but it's a very important process that involves the development of assays, screening, biomarkers, and drug discovery. But indeed, you know, they work hand in hand. You have this is just simply the platforms that we use. Everything is available to inform on mechanism of action, going back to the hallmarks of cancer. We've developed and leveraged systems that allow us to inform on every aspect of the cancer biology paradigm. Not going to go over this. So lead ID, this is when we start the fun stuff. Now you have a target. You validated that target. You show that it's a maintenance target, meaning that if you extinguish it in an established tumor, the tumor shrinks, so stops growing. Now comes the most difficult part. Generating a compound. Identifying a compound. So what we need for lead ID, right, for a small molecule, is to develop a minimal screening funnel, and I'll talk about this on the next slide, to identify novel chemical matter. Identify an inhibitor that binds to the target and inhibits that target, and it's optimized for potency. Potency meaning that it is potent against that target, that you need very little compound to inhibit that target by 90, 100%. And so this is really the screening funnel, and you should have this in your thing. There are 3 components, right? There's an in vitro biochemical or biobinding assay. So we have our target. We're going to study microphone, protein microphone, right? And so this microphone is an enzyme, and it phosphorylates this podium, right? And so what we would do, it's a validated target. If I take the microphone away, the podium's worthless. How's that? Coming up with all kinds of analogies today. Right? So the microphone itself, what we would need to inhibit this, or simply shut it off, is not necessarily a button, but a small molecule. And so you would need a biochemical assay. You need an assay that you can test the activity of the microphone with respect to the podium. And that if I were to implant a compound on top of the microphone, it would inhibit the activity, right? You need a cellular target engagement assay. Now my analogy becomes very difficult. But a target engagement assay would be the activity, the downstream activity of the podium. And then a phenotypic assay, the fact that you can hear me through them. This is a terrible analogy, actually, but I put it together. How about we just go back to gene A and substrate B? Yeah, let's just inhibit that whole aspect. But the idea is to do this, is to inhibit the enzyme in a test tube, inhibit the in a cell, and then see what the functional consequence is. And so you have in vitro, which is the enzymatic assay. You have the cell biochemical. That's the target engagement. What is that enzyme doing? So if enzyme A phosphorylates substrate B, we would measure substrate B phosphorylation as a target engagement biomarker. And then you have cell functional. What is the phenotype? So lead optimization. This is when it becomes critical. Because you can have compounds that inhibit your enzyme, but would never work in a patient. And so again, that's when we shelve it. We kill the series. Work on something else. Go back to lead ID, right? This is where you install all the desired properties of a drug into a single compound. Now this is very important, because this is when you come to the realization that the chemical series you're working on could work in a patient, at least function in a patient. This is where you talk about one, PK, pharmacokinetic properties, what the body does to the drug, right? We talk about distribution, we talk about excretion, metabolism, you know, the liver, does it just chop this up and send it right to the kidneys? You urinate it out within minutes, right? Pharmacodynamic is what the drug does to the body, okay? PK, what the body does to the drug. PD, what the drug does to the body, all right? And so solubility, will it cross the gut, right? If you take a pill, will this be absorbed into your bloodstream? Plasma protein binding. Chemistry loves proteins, loves binding proteins. You have a lot of proteins in your blood. So if 100% of your compound is binding proteins in your blood, it's never going to get to the desired site, the tumor, right? So plasma protein binding. Hepatic clearance, getting it through the liver. Drugs are metabolized in the liver. They have a number of cytochrome enzymes that do that. That's their function, right? Renal clearance. Can we at least get it through, you know, the kidneys and the renal system? And then you're talking about tissue distribution, right? If we can get it through if we can get it absorbed, get it cleared through the liver. If you have a tumor in the brain, does the compound go to the tumor in the brain? If you have a tumor in the colon, does it go to the tumor in the colon? Breast, lung, everything else. And so those are the type of things that we need to inform on. And we do this very simply, right? We use the mouse models. We feed the animals through gavage. We put a needle directly into their stomach, right? We empty our compound into their stomach. Then over a period of 30 minutes to 24 hours, we take blood samples, we take tissue samples, and by using mass spec, we can say, all right, how much compound is left in the blood? How much compound is left in the tumor? How much is in the brain? And so this is our PK, what the body does to the compound. If you have a terrible PK, and the half-life of your compound in blood is 10 minutes, kill it. Move on to another series. You need something that's going to work. You can't necessarily have a half-life of 10 minutes and then tell a patient, okay, you have to take 32 pills in 24 hours, right? It's quality of life. You need something that you can manage.
Delivery directly to the stomach, and not orally or bloodstream-wise.
So absolutely. So there's a number of ways. If it doesn't pass the gut, and there are a number of great drugs that don't. They're not taken orally. That's when the patients go in for outpatient clinics and they get transfused with their chemo, right? So there are a number of different ways to do this. But when you talk about quality of life with a patient, if you can have an oral bioavailable drug where you can say, go home, just like our, like Lipitor and other drugs that people are taking, and say, I'd like you to take 2 pills every morning for the next 30 days. That's a lot easier than saying, I need you to come into the city, I need you to come into the hospital, you're going to sit down for 4 hours, we're going to infuse you every day for the next 30 days. So it's just, you know, how do we manage this? How do we make it as easy as possible on the patient? But again, it's not necessarily the route of delivery that's the problem. It's what the liver is actually, and other organs are doing to the compound. We have to make it available. So then we go into this process called SAR, structure activity relationship. And so the first compound that you identify through lead ID is hardly ever your clinical candidate, right? You go through this iterative process of screening. So you have efficacy and selectivity, right? Is compound X potent against kinase A? You get information. Yes, very potent, very selective. And then you go through design, you build more compounds, and you go through this process. And then when you get through information, you go to compounds. If this passes, now you have a compound that's very good. You pass it through your preclinical models. You get kinetics, metabolism, ADME, DMPK. Then you get more information. So you go through this iterative process that you make changes, small changes to a molecule. You change a hydrogen to a nitrogen. Well, now it doesn't work. Okay, you throw it out. You go through the next structure. You change that hydrogen to an oxygen. Oh, now it's tenfold more potent. And so we go through this iterative process. This is why it takes so long. It's not necessarily target ID. I could give you 10 targets tomorrow. But it's identifying a compound and then making that compound potent, selective, that'll work in a patient. So this is a long, iterative process. You know, a program that we're working on, we had a compound currently working on that we were able to file patent within 8 months. From lead ID, we identified a compound that was a single-digit nanomolar inhibitor, which is incredibly potent, right? We are still running SAR to inform on the clinical candidate. So day one, we had a 1 nanomolar inhibitor. So now we're 12 months in, and we're still running SAR. So you can see this takes a long time. So then there's development and translation. So you have your target, you have your chemical, your probe, and you need proof of biology. And so here, we don't necessarily do this in the patients. We want to develop this using biomarker-driven studies. Robust translational strategies, as I say here. Effective agent to patients likely to benefit, right? Can we stratify this room? Again, I use this saying better than the microphone. This analogy is much better, right? 10 people over on this side, 5 people over on this side have the biomarker that I desire. I have made a drug that'll work for those 15 people. Instead of giving it to everybody and watching, 55 of you not benefit, I'm only going to give it to the 15 that I know will benefit. Okay? So that's a very different way of thinking about drug discovery. That we do this up front. We're not going to do this in clinical trials. We're going to go to the clinical trial and say, you need to treat this population of patient. And this is where this type of early development, preclinical development, comes in. And this is what it takes, right? Preclinical testing in the appropriate model system. So in your genetics books, you'll probably read about a number of these things. This week, you'll probably hear a number of these things. Some are more robust and simple, cost-effective, high throughput. Some less so, but more informative, more costly, slower. Those include cell line panels, like I talked to you about today. We can take 500 cell lines and treat them with 10 compounds in a matter of 3 days, right? Robotic, with automation, of course, but that's very easy. But using genetically engineered mouse models takes months, a year, to test efficacy. We can do simple in vitro assays. We can use patient-derived cell lines. We can use cell lines, what are called patient-derived xenograft models. I'll show you in the pancreas. In the end, we want to use whatever model will help us predict what patient in a phase 1 trial, phase 1b trial, will benefit. And so we utilize everything and anything that'll help us inform on a strategy for the clinic. This is one approach. This is from a good friend of mine, Jason Fleming. He's a surgical oncologist at MD Anderson. This is a pancreas cancer. So this is the pancreas. This is the cancer, right? Very deadly disease. Clearly, there's an unmet clinical need here. Pancreas cancer needs a new therapy. So what he does is he resects part of this, just like we do in GBM, resects part of this tumor, dices it up on plastic for just a few minutes, implants it into a mouse, and then we grow the human tumor again. That allows us to increase the amount of tumor that we have to analyze, that we have to work with. And as you can see here from histology, this is what the patient's tumor looked like. And on the right-hand side is what the tumor looked like after we grew it in the mouse. Looks very similar, right? And so this is probably as close to as you can get to the patient. And so we can use these PDX models as preclinical models. If it works here, Will it work in the human? We don't know, but the chances are better. If it doesn't work here, it's definitely not going to work in a human, okay? And so these are the preclinical models that we talk about. We also use genetically engineered mouse models. I'm not going to go into this too much. This is a melanoma model, right? We can form melanoma. We can extinguish RAS. We can make those melanomas disappear. This is a very complicated study on this slide. Very complicated. I'll sum it up in just a few sentences. We used a mouse model, right? If we extinguish RAS in this model, genetically, the tumors regress. However, if we treat with available compounds that are in preclinical development, like a MEK inhibitor, we see stasis for a while, but they relapse. But we never see this type of extinction. So what we did, this was done by Larry Kwong and Linda Chin, was to use network biology, to use genomics, transcriptomics, using microarray and proteomic data from reverse phase protein arrays, inform on a RAS-specific module. And through this, inform on a combination treatment. This is a long story short. This is a great paper, but in the interest of time, we won't go into it too much. But through this network analysis, this cotton ball on the top right, there is an association between MEK extinction and CDK4, a cyclin-dependent kinase. So it informed on a combination treatment that included a MEK inhibitor plus a CDK inhibitor. So through this study in the mouse, through what we've been able to show, using this mouse model, Novartis is now accruing patients for a phase 1b, phase 2 trial, combining a MEK inhibitor with a CDK inhibitor in NRAS mutant melanoma patients. So you can see, granted this was several years of study, but using a genetically engineered mouse model, we've changed, we've basically initiated a clinical trial in big pharma. And so that's where these preclinical models come into play. Could we have done this with a number of other different models? Probably. But what we've done here is integrate a mouse model of human cancer and functional genomics, transcriptomics, integrated genomics, and informed on a rational drug-drug combination. And so these preclinical models are very useful. So when talking about, and I'm almost done on the next couple of slides, but when thinking about MD Anderson, we're starting this translational continuum, right? I told you earlier that to make a drug, it takes 12 years, $1 billion, and after that, only 5% are actually good. They pass everything. So what we're trying to do is accelerate this process. Accelerate the process of drug development from target ID to clinical candidate, and then improve the success rate in the clinic by simply informing on which patients are likely to respond. So you have target identification and selection, validation of the target, in vivo efficacy studies. Once you have your tool compound, use the appropriate preclinical models, right? Validation of disease indications. You have your target. Will it be melanoma? Will it be GBM? Will it be colon? Say we have colon. Are we going to treat all colon cancer patients? Or are we only going to treat KRAS mutant or BRAF mutant or EGFR mutant colon cancer patients? Biomarker strategies for patient stratification. This is the key. 10 people on this side, 5 people on this side. Instead of taking all of you and seeing 65 of you not benefit from this drug, we're not going to waste our time. We're not going to waste the money, right? We're not going to waste your health. We're going to take the 15 people that have the biomarker we desire and put them on our trial, and they're going to benefit. The other 65 of you have a different biomarker. And then we may subdivide you into 20, 20, and 25, each getting 3 different types of drugs. But right now, we need that preclinical, the preclinical data to prove that. Validation of early response, right? What happens in a clinical trial? You get treated for 6 months. You show partial response. They take a biopsy. What we want to do is do this in a matter of weeks, right? If it's not working, we don't want you to take the drug, right? You have the biomarkers that'll predict response and efficacy. If we're not seeing it in a matter of weeks, if we're not extinguishing ERK phosphorylation as a distal biomarker or proximal biomarker, then we'll take you off the clinic. We'll take you off the trial. We'll give you something that you might benefit from. Not you. I don't want you thinking that you're eventually going to be patients, but I can see some scared people in the audience. But the reality is, this is the reality, right? This is what we face in oncology. Early phase clinical trials, right? Yeah?
How do you adjust the doses of the trial?
The dosage of treatment?
Yes.
No, that's, if your phase 1 trial is always going to be the same. Your phase 1 trial has to be dose escalation, right? And so we know that through preclinical studies that you would need to inhibit the enzyme by 80% to see efficacy. And so in a phase 1b trial, what we do is we treat a couple patients at 1 dose. We early, detection, we see what the response is. If there's no dose limiting toxicity, we'll increase the dose. We'll have another 3 patients that are 4 times the dose. We'll keep increasing until we get what's called the MTD, or maximum tolerated dose. And so that's when you start seeing toxicities. At that point, you've reached it. And then we test preclinically what the response is. But we should know that. At least the MTD, you can't guess what the MTD or calculate MTD from a mouse study. But what the mice can tell us is the response and efficacy that we would need, or the response we would need to achieve to see efficacy. But it's a great point. But we can do what are called co-clinical trials. We're building a center right now where we're using human tumor samples implanted into the mouse, treating with compounds, diverse compounds, not just ours, many compounds, to try to inform on biomarkers that would predict response. Do this in the mouse. Let's not waste a phase 1b trial on 100 humans. Let's use the preclinical models that we have. To do this, we have dedicated animal facilities, right? So all the imaging I've been showing you, CTs, MRIs, CAT scans, everything, right? We have this available for mice. So we can treat the mice and that individual mouse like a patient, right? And so we can inform on the genomics, genetics, the context, I come back to that word, that are likely to respond. So again, this becomes the whole process. This takes an integrated effort. As you can see here, this is a grid. We go from target selection, lead ID, hit to lead, all the way through phase 1-2 trials, right? But it takes an integrated effort. It's not a team of 4 people. It's integrated with target biology. Target biology can inform on responder ID, can inform on biomarkers, can inform on proof of biology. And that helps the chemistry. But as you can see here, there is a project team. I run 2 drug discovery programs. The people who report to me, there are 4 people. 4 people on 2 drug discovery programs. But because of the matrixed organization that we've built, it's fully integrated. So my project team, my biology team is 4 people. But each project team is about 20 people. We have a biomarker group. We have bioinformatics, computational biology. We have computational, medicinal, structural chemistry, right? We have 5 chemists working on my program. In-house. We have another 5 outsourced to China. Mouse models, preclinical models, oncogenomics, molecular profiling. This is what it takes to run a project team, to run a drug discovery biology program. You need all of these parts. This is not necessarily to prove to you that we have everything at MD Anderson to do this, but just to prove to you this is why it's so hard. This is why it takes so long. That a group of 3 or 4 people, it's not going to be very easy for them to execute on this. This is the type of activities that you need in a drug discovery program. What I've listed here, and this is a little bit outdated, everything is in blue is what we have in-house. Everything in green is what we do in-house but outsource. And red is external, in which we utilize the outside world. Biochemistry, enzymology, cell biology. This is all, not to, beat the horse here, but this is what goes into drug discovery. I think it's truly important to understand. But what we have is access to and the ability to leverage the best and latest science. We're integrated in an academic center. And we're committed to science-driven decisions. Again, we can call a no-go decision. We can say, it's not worth it, let's cut our losses. We invested $3 million, but we just did this killer experiment and it didn't work. So you put it on the shelf. You know, we have a program now that we're, you know, by the end of the year, we'll have our preclinical candidate. Right? If something happens between now and then, it'd be 2 years. It'd be about $7-8 million that we have invested. But we would rather stop there and cut our losses than go into a phase 1B trial and show that the patients don't respond or it's not safe. And then we lose 30 or 40 or 50 million. Do you know what I mean? And so it's critical to perform these preclinical studies. And this is really the idea here, that there is multiple go-no-go decisions. There's target validation, context specificity from the beginning. You know, what patients would likely respond to if we extinguish this target. Stringent loss of function studies. We got to inform on tumor maintenance. Pharmacological target validation. I told you we use tool compounds, chemical probes. Develop a minimal screening funnel to identify the compound. Proof of concept screening. Do these compounds work? Efficacy in vivo, animal models. If your biomarker says BRAF mutant melanoma, and you treat a BRAF mutant melanoma mouse model, and you see efficacy, great. If it doesn't, you're done. It's a no-go. Clinical path validation. What do the human genomics tell us? And lastly, biomarker discovery. It's not just predictive biomarkers. It's not determining which 15 of you we would treat. But it's also efficacy biomarkers, proof of biology. There's proximal biomarkers, phosphorylation of substrate B. There's distal biomarkers, proof of biology. Do they stop growing? Do you see increased DNA damage? Do you see metabolic adaptation? Those are all things that we can quantify. And this is the difference. This is what, you know, at MD Anderson, what we're trying to build, which will basically provide a package to Big Pharma, a data deck to say, this is worth it. You know, this target, this is all of our preclinical data. We have models that reflect genetic alterations. We have monotherapy or combinations. We have biomarkers, predictive, but also efficacy. You know, if you give this drug to this patient population, you will see response. And that's going to improve the success rate. And we can best do this in an academic setting at MD Anderson. We do not answer to shareholders, right? We don't answer to, you know, the next quarterly update on Wall Street. For us, you know, our motivation is to make drugs. And so for us, we can take advantage of every opportunity in academics and industry to do just that. So let me take, I think I got 3 slides left. One of them's an acknowledgement slide that we can ignore right before lunch. So just to give you an idea of what's ongoing at MD Anderson. I think this is very important. It was all over the news. So Ron DePino, who was named president of MD Anderson about 1.5 years ago, has decided that the time is now and to build what's called the moonshot platform. So in the 1960s, John F. Kennedy, who was the president of the United States, spoke at Rice University in Houston, Texas, and basically said that the United States was going to go to the moon, right? Thus the moonshot idea. So this was 50 years ago. And so back in 1962, when we were talking about going to the moon, we had all the technology, we just didn't know how to use it. And so that's the analogy, a much better analogy than the microphone and the podium, right? And so this is the idea that we have all the technology to conquer cancer, reduce mortality rates. We just need to work together, integrate all the activities, from genomics, functional genomics, all the way through drug discovery, biology and chemistry. And so these are the 8 inaugural programs. The goal is, through funding in upwards of $3 billion over the next 8 to 10 years, we will reduce the mortality, triple-negative breast and ovarian cancer, leukemia, AML and MDS, leukemia, CLL, lung, melanoma, and prostate cancer. Those are the inaugural programs. There are other programs at MD Anderson, that are not full moonshots, but sort of, this is way corny, but it's the only way I can explain it, is like orbiting the moon a bit, right? So just flying around it a couple times. And so they need a little bit more data, or, you know, a little bit of help before becoming, you know, being able to land. But nonetheless, this is the activity that's going on in MD Anderson. And so when you think about the platform, you can see that this is an integrated effort. And it's not just treatment, right? It's not just coming up with the next drug, next targeted therapy, but it starts with prevention and early detection. Prevention, not going to the sun tanning booth, you know, tobacco, you know, things like that. Prevention, early detection, imaging technologies that we could detect an ovarian cancer or prostate cancer or a lung cancer much earlier than we do when a patient is symptomatic. Omics profiling, immune monitoring, harnessing the power of the immune system. That is probably the most advanced, well, the most exciting advancement in the field of oncology through Jim Allison and others. Functional genomics and systems pharmacology, identifying those targets. Big data, using artificial intelligence to predict response. Drug discovery, that's where we come in. And then the translational research continuum, the preclinical, co-clinical trial center, using human tumors implanted in the mouse to predict before we go to phase 1. So that's what we're doing at MD Anderson. This is where we are in red. The I-axis really spans everything from target ID to clinical development, but we're leveraging the MD Anderson community, academic community, the TRC, but also the individual moonshots. So together, it's a group of, you know, almost 20,000 employees at MD Anderson, from clinical to basic science, and most importantly, the patients that allow us to learn from you know, the hardships that they've had to face. And so through this, and so when you think about genetics, it's not just a linear pathway. Think about genetics when you're in your class. Think about the impact of genetics. And it's beyond simply whatever the guys that experiment with plants. I can't even remember his name. Gregor Mendel. It's more than whether the flower is green or yellow, right? That it impacts human disease pathogenesis. But I appreciate, you know, it was all, I don't know, I had a lot of fun. I hope you learned something today. But, you know, the near future and this, I promise, is the last slide, and then you can eat. But I told you earlier about the problems and the challenges that we face, right? This is the near future. I wouldn't even call it the future. This is happening. A complete compendia of cancer-related targets, right? In 50 different cancer types, every subtype within those cancer types, and every subtype within those subtypes. Disease modeling in the appropriate context, right? Tumors are neo-organs. That, you know, motivated me to go in vivo as soon as possible with these screening efforts. Let the organism tell us. Understanding context, biomarker-driven clinical trials, right? It's not going to be all comers, stratified. Improve compound quality, right? We are doing better chemistry. Earlier disease setting for most trials. Higher risk tumors identified at the earlier disease stages. So we're not dealing with a stage 4 that has metastasized throughout the body. That maybe we're dealing with, you know, one driver that we can attack. Imaging, safer compounds. But this is, you know, the introduction of genomics, functional genomics in the I-axis, but that's really where we're positioned after moving to Texas. I don't want to go into any acknowledgments, simply to say this is our building and the 70 dedicated scientists and associates that help do all the work that I get to present. But I appreciate your time. Again, I'll spend some time at lunch, and if you have any questions, and appreciate the opportunity to talk to you today, and enjoy the rest of the program. Thanks.